Okay, looks like the sign-ins are starting to slow down a little bit more, so we can get started. Hello, welcome to today's CCHAB meeting. We have our exciting kind of first meeting of the year where we go over what last year's conditions were like. Um, oh, yeah, don't worry, Thomas. I know that you guys can't access, if you can't access Google Docs, if you wouldn't mind just throwing your names in the chat and where you're, I know where you are, Thomas, but where you're from, and I will manually record that for you guys. Sorry, I know we don't have one system that's compatible for everyone, unfortunately, but thanks for being here. So welcome to the CCHAB meeting. If you haven't yet, please go to that Google link in the chat and let us know that you are here. And I wanted to introduce myself. I'm Karen Odkins. I'm at the California Department of Fish and Wildlife. I'm one of your co-chairs at CCHAB here. We also have other folks. Go ahead.
Yeah,
Cool
I'm
chat.
Jamie Smith. I'm one of the co-chairs. I'm a senior scientist at SWRCB.
Great. And Sarah, are you? She might be transferring computers right now. Okay. Yeah, so we have Sarah Ryan as well from Big Valley as one of our co-chairs, but she will be joining us in a minute. She is bringing people from the other link that we previously had. Okay, so let's go into our agenda today. So today we have an awesome theme of mitigation, and we have some great speakers, 3 different research presentations in addition to all of our updates from the folks at various regions around the state and also from our subcommittees. But I'm going to first start with some quick announcements that we have here today. So let me share. Can you guys see this window? Someone unmute and just let
Yes.
me know. Yes? Okay, awesome. Thank you. Okay, so one exciting thing that's happened is that we have an updated website. And so I wanted to show you guys what it looks like and just give you a little tour really quickly. You guys can explore it on your own as well. But we have this homepage here that now kind of lists what we do and links you to various places that are common people or places people are interested in. And then, you can go straight to the meetings and you can also see here when our upcoming meetings are for the calendar year. And then you can click on our calendar as well. And that'll get you to our agenda. And also you can look at all of our past meeting recordings and minutes and agendas for all these various years. So that's on the homepage. If you click on About Us, you can learn about the Monitoring Council and what we do. You can go through our membership, and this page I think we're going to update soon based on all of these responses that we're getting from this attendance survey. So this is kind of older information, but will be updated soon. We also have information on our subcommittees, both the active ones and who to email if you want to join them, as well as some of the previous ones. So you can see all the cool things that we've done in the past. And then also, who you can talk to connect with us. And also, if people ask how to join and want to get on that mailing list, this is the place to send them to for email subscription to our mailing list. This meetings link goes straight to that meetings page that I showed you earlier. And then I'm going to skip over the HABS portal right now because it goes straight to the HABS portal, which opens in a new tab. But if you want, it's kind of that connectivity between the state program and the state board program and the CCHAB website. And then we also have a resources page, which goes through all the various products that CCHAB has produced, and then also some of the most popular resources that folks want to get to. And then, of course, we have our report a HAB button, since that's something that people, want to quickly get access to. So just wanted to share that with you. If you guys have any suggestions as you're going through it, this is not a static website. We can change it as needed. So feel free to email me if you have suggestions, and we can see if we can pull those off. Um, Great. And so I think we will transition into our FHAB or our updates from various regions. So starting as we do with region one, or actually with state board, sorry. First, are you presenting with that? I can click through slides for you.
Yeah, this is great.
Really.
Thank you. Okay. Yeah, you can go to the next slide. I'm Carly Nilsson. I work with Marissa Van Dyke at the State Water Resources Control Board. And yeah, you can go to the next slide. Shoot, I think. Oh no. Froze. What
Is the
happened?
wrong slide, Carly?
Okay, I'm back. Can you hear me now?
Yeah, I can hear you.
Okay, I don't know what is going on with my computer. Okay, you can go to the next slide. Am I still here?
Yeah, this
Okay.
is the reminder CCHAB. Is it delayed on your side?
Oh, yeah, which are you on slide two? Oh, here we go. Okay, sorry. Guys, I don't know what's going on. I'm having technical difficulties, obviously. I'm gonna turn off my video because maybe that'll help. Okay, so this is just a quick reminder that we have new CCHAB guidance for our benthic cyanobacteria sampling methodology. It's a little more involved now with including percent coverage. So please, if you're not familiar with it and you didn't take one of the trainings last year, reach out to us. We can do one-on-one trainings, point you to the resources. We're actually getting a lot of help internally of getting like a really great library of resources, including videos. So just a quick reminder, you can do that QR code to kind of start scoping it out, but don't hesitate to reach out to us. Next slide. And the FI coordinators are going to go into this in a lot more detail, but we just want to give a big, huge thank you to all our partners and our FF coordinators who did our pre-holiday assessments this year. We had quite a few, including quite a number of sites that were posted with public health signage, including 30 partners, 4 tribes, and as well as every single regional board participated or trained partner. So we have just grand appreciation, and we hope to see you all back this year. Next slide. Okay, so we have some cool new remote sensing things. I think we've brought up some of these in the past, but I just wanted to give an example of like how impactful this can be for folks, especially for these larger water bodies. So our Shiny app can be accessed through our SFEI remote sensing map. I think Marissa can throw a link in there for you so you can get there quickly, but this is just focusing on like Elsinore, and this is looking at bloom magnitude. And we just want to highlight like they've been doing a lot of really cool mitigation work there. And you can really see the differences in the bloom magnitude in 2025 based on that. So it's a really great way just to like track how, mitigation or management measures are working on the ground and use some of these remote sensing tools to really actually visualize those differences. So I just wanted to highlight that. It's a resource, it's available, it's to the public, and you can look at frequency, occurrence, and spatial extent. You can look at water bodies, you can look at things regionally. So there's a lot of different ways to check it out. If you have more questions, as always, you can reach out to Marissa and I. We're happy to help you. Next slide, please. Okay, so talking about some things that are on the horizon. So this year we're going to be doing a pilot benthic cyanobacteria ambient monitoring study. We have a technical advisory committee convened currently, and we're kind of scoping that out at the moment. But it's really going to be an awesome project to scope out how we could get like a really strong ambient monitoring program across the state for benthics. That's something that we're still sort of a black hole for us in California. I know the North Coast has done a ton of work and we are extremely grateful for that. But in terms of other areas across the state, we still have a lot of unknowns. So we're scoping that out. If you have interest in it, please reach out to us. We are maybe looking for some additional participants this summer. It would be like a pretty firm commitment through June through October. And probably monitoring at least once a month. But it'd be a really great way to like dive into a watershed and really see what's going on there. And then as well, we started this last field season and kind of got some of the scoping out and got some of the documentation together, but we're comparing different sampling methodologies for sampling for benthic cyanobacteria, including the CCHAB network, new SOP, standard operating procedures for collection, and we're evaluating that in tandem with like Utah's percent coverage and then US EPA's draft methodology, which also uses a transect method similar to Utah, but also collects cyanobacteria for toxin analyses. So we're just trying to make like really hone in our bacteria or benthic cyanobacteria monitoring program to make sure we're being protective of human health and our doggy friends that are in our state. We're going to continue to prioritize HAB incident response across the state. That is mandated through our program, and we feel very strongly about it. So a lot of the counties and water body managers, you hear from us often in the during the HAB season. So you will continue to hear from us, and we will continue to provide support as we can with sampling and/or analyses. So we really look forward to those partnerships this summer. And then we're going to continue on with our pre-holiday assessments for 2026. So keep a lookout. We're going to be doing a solicitation for folks to participate with us. As you can see, it's a really strong program, and it really does provide that preemptive data so that we can get that signage out before those big holiday weekends to keep folks safe. So it is our most robust program for public safety at the moment. And next slide. And maybe it's just not going for me. Oh, here we go. And this is just another plug again. Reach out to us. We hope to hear from you. It's a really great time right now to start training if you have an interest, or if you know you're going to have HABs again this year and you have new staff, or if you have monitoring plans you want to start designing, if you have some management programs that you're trying to get on the ground, and just making sure you're ready for next year. You know, we can provide sampling supplies and trainings and different things of that nature. And that way you're just preemptively prepared. So that way we're not coming to you when it's potentially like incident response and everyone's sort of scrambling to get on the ground. So we just want to make sure everyone across the state is prepared and ready and ready to act. So don't hesitate to reach out to us. We love those one-on-one trainings and that dialogue. So we welcome it and hope to hear from you. And with that, I think I'm done.
Great. Thank you, Carly. I think, Marissa, you were going to cover for Mike at Region 1.
Yeah, that's right. Hey, Karen. All right. Hi, Stacy. Have folks. Yeah, I'll be covering a few of the FAD program coordinators and supporting them while they're out in the field today and providing their grand updates. I'm Marissa Van Dyke with the FAD program, colleague of Carly Nelson, who just spoke. And so this is the update from the North Coast region. Thanks for putting that map there. So just to get you guys all oriented, it goes from Siskiyou County, Del Norte in the north down to Sonoma. And this was put together by Mike Thomas, the regional monitoring coordinator. Next slide. All right, so they'd like to highlight some of their routine monitoring projects that span the past year. There's 3 really cool projects. So one is in the Klamath Basin. This was implemented along with the Tribal Consortium that's been also monitoring the Klamath Basin for the past 16 years. So this was before the multiple dam removals of the Klamath River. So the Tribal Consortium has been monitoring for a lengthy time. So it's a really great partnership to look at monitoring of benthic gnats. In the past, they had been doing primarily stream monitoring from planktonic blooms in the reservoirs. So this was a new data set to collect. Also, the second project was in Big Lagoon. This is a coastal lagoon. They have been partnering with Big Lagoon Rancheria to do biweekly microsystems monitoring for planktonic blooms. And then the third project is in Russian River, and this is biweekly sampling for benthic blooms by the Sonoma Water District and the county, that kind of partnership there to also implement spot monitoring, visual indicators and that. So replicating the ambient monitoring protocols in the benthic SOP from CCHAB. Next slide. All right. So we'd like to highlight the partner monitoring project in the Klamath Basin this past year. Again, this was tribal consortium, including the Karuk tribe, Yurok tribe, Hupa tribe, and Quartz Valley community as well. And so they implemented a combination of using SPAT deployments, that's that passive sampler, visual indicators, MAT collection, and also qPCR for toxin genes. Again, that's that ambient monitoring protocol in the CCHAB benthic SOP for the field. And the data is going to be wrapped up in a new report that will be shared into the CCHAB listserv. But we're just getting like a little preliminary data set here. And next slide. All right, so there are 12 stations that were monitored from approximately July through November as part of this project. And the left graph here is showing the antitoxin concentrations in the SPAT bags that were deployed at each of these sites. And it's highlighting when the detections exceeded this threshold that is presented in the CCHAB SOP as well. And that threshold, when exceeded, it triggers next actions, including postings as well. The right graphic is showing many things happening here. So this is, again, the data set of the SPAT, benthic, MAT, and qPCR data spanning from July through November. We're showing on the left-hand side that the SPAT concentrations, and then the right axis has the qPCR and a toxin gene as well. And what we're trying to show here is the results from the field collections correlated. And this is showing that there was correlation between the SPAT detections that were deployed before the benthic MAT collections. So that's something that has been observed in previous studies as well in the North Coast. And we're seeing that, the qPCR results is something, using toxin gene detection is something that we're still researching. And so it's really interesting to see all this data set together, and that will be coming out in a report. And through this study, there were 5 time points where postings were triggered. And this included, several tributaries of the Klamath River as well, and that including the Scott, Kidder, Salmon, and Trinity rivers. Next slide. All right, so going out of the studies, and we wanted to highlight some of the pre-holiday assessments data and those results. So in this region, they elect to do a combination of the pre-holiday assessments before the major holidays, Memorial Day, 4th of July, and Labor Day, and also a post-holiday, depending on how the blooms had started developing at the early part of the year. And so this is showing when the holiday assessment sampling resulted in a posting for recreational advisories through these different kind of water bodies, mostly reservoirs and coastal lagoons, here on the left. And really couldn't do this without all of our partners, so we wanted to highlight that as well. Next slide, please. All right, and then the posting, additional posting decisions were made in combination with bloom reports that were voluntarily submitted and along with the holiday assessments. So this is showcasing on the top the benthic advisories, or benthic cyanobacteria bloom advisories, and the planktonic bloom advisories. And some of the alerts were for toxic algae alerts were in many of the streams in the North Coast region, and some of the highest planktonic advisories were Big Lagoon, Lake Pillsbury, Lake Shastina, and Big Lagoon. Next slide. All right, and they also like to highlight the illness reports that were submitted. This is, again, with that bloom reporting tool that we have on the HAB portal. You can also submit if there's a concern for illness of yourself, your family, also pets, livestock, and fish and wildlife. And so this is the results of that reporting. There were human illnesses, dog deaths, and fish illnesses reported. And there's different determinations depending on the illness work groups, case determinations in that investigation. So we get many suspected illness reports, and depending on the course of investigation, it may or may not be determined to be HAB related. And so that information here is presented on the right for the determination. Next slide. All right. And some accomplishments from the past year, we're doing several training sessions. Of note, there were 3 different options for hands-on field training through the North Coast region in August that really helped prepare many partners to do benthic monitoring and also like recalibrate folks that are familiar with benthic monitoring. So this was really a great opportunity to get out there and get everyone geared up for their monitoring and any of that response. And there are virtual recordings available as well if anyone would like to see that. Next slide. All right, and plans for the upcoming year is reassessing those same lakes and coastal lagoons, 7 water bodies in total for the pre-holiday assessments. They're going to be participating in the statewide special study looking at benthic ambient monitoring in rivers and also assisting partners with establishing their own monitoring programs. And there's plans to do these field trainings and virtual trainings again, so please stay tuned in the listserv. And we are looking forward to bringing on a new FPAB coordinator this year as well. All right, that's the end of it. Thank you.
Thanks, Marissa. All right.
Region 2?
Yeah.
All right. Hi, this is Christina Yoshida. It's always so hard to go after Region 1. So many awesome things. So actually at the last meeting, I gave our year-end summary. So I didn't want to bother you all with the same information again, but just kind of in general, it was a pretty calm year for HABs that were reported to the Water Board. We did partner with 3 community groups to help collect our samples for holiday assessments in Napa and Sonoma, and also in Lake Merritt in downtown Oakland. And we didn't have any advisories linked to these community group helps. Again, we're lucky to partner with East Bay Regional Parks that shares their weekly monitoring data, and I know we're going to hear from them later. I know this year we also made some connections with other local agencies and groups that will now be sharing some of their data for our advisory maps like MidPen. And again, luckily I have Kevin Lundy, who's now on board with HABs, and he's focusing on the San Francisco Bay. And so he's going to give an update on Bay HABs and also Lake Merritt. All right.
Sounds good. Thanks, Christina.
Yep.
Hey, everyone. So for San Francisco Bay, just a reminder that we had no kind of major HABs over 2025, which was good news. There was a kind of a small HAB along one margin of the bay, and appreciate working with East Bay Parks on that. In terms of the bay, just another kind of reminder on the monitoring program that we have. We have the SFEI as tasked with managing the nutrient management strategy, which is kind of a discharger-funded nutrient monitoring program, and it collects chlorophyll A, phytoplankton, dissolved oxygen, nutrients through various mechanisms all throughout the bay. It's a pretty robust program, funded at about $2 million a year, and thankfully this year we were able to get additional money from EPA to fund kind of a multi-year expansion of the program and actually increase that funding for a couple of years. So we do a lot of satellite monitoring as well with that program, and they're expanding the tools, and maybe in a future kind of meeting we can summarize some of the tools. They've developed some great shiny apps that let people kind of explore their SON data or explore the satellite data or other data sets that have been collected. And then also to let folks know, kind of I put it in the mitigation umbrella, is that because of the large heterosigma Akashiwo bloom in 2022 that resulted in widespread fish kills, the Water Board required nutrient reductions throughout the wastewater treatment plants that discharge to San Francisco Bay, or that maybe discharge to rivers that discharge into San Francisco Bay. So it's a widespread reduction. It's 40% reduction. Because this is a big change, it takes a lot of time for wastewater treatment plants to plan, kind of consider their options, and then do environmental review like CEQA on their approach, get all their necessary permits, and then construct. So at present, they have 10 years to do all of these large-scale reductions, but we are aware that certain projects like nature-based solutions, such as like horizontal levees or open treatment wetlands, uh, will actually take a little bit longer. So we're working on a basin plan amendment to give certain projects more time than 10 years to comply with that nutrient reduction. And happy to chat more about that with folks if they have follow-up questions. And then for Lake Merritt, we're doing it's kind of what's called a TMDL alternative in Lake Merritt to manage low dissolved oxygen levels. So we've got a bunch of sondes out there and people monitoring Lake Merritt, and we had a report of a harmful algae bloom in December, which is not our typical window, but it was helpful. We had some people on the ground right away who could collect a sample. CDPH could do the identification, and it turned out to not be a harmful algae species, and the bloom dissipated in a couple days. It did result in a shift in DO, but no decrease. So thankfully, it ended up not being harmful to kind of human health or aquatic life. And that is the update from Region 2.
All right, thank you. I believe we have East Bay Parks next.
Hello, can you hear me?
Yeah.
Awesome. Okay. My name is Ikayla Autry. I am a water management technician with the East Bay Parks. And we had a fun 2025. This image, though, is our most recent HABs advisory as of January 9th. So Quarry Lakes, Lago Los Osos, and Shen Pond are at dangers, and Lake Del Valle boat launch is currently out of danger. And every other site we have, with the exception of Shadow Cliffs Arroyo, which is like a frequented dog water body, and Big Break. Every other site besides those two has a caution advisory right now. You can go to the next slide. Oh no, did my animations? You might need to click it.
Yeah. One more.
Thank you.
Good?
Yeah, that's good.
Okay.
Okay, so how did we do in 2025? This graph just shows, as many of you can see from the colors, that for the majority of the swim season, our swim beaches had advisories. Shadow Cliffs remained at a caution the entire season because despite getting consistently low microcystins toxin results, it had consistent benthic cyanobacteria presence. Quarry Lakes had the longest amount of danger advisory days out of all of our swim beaches and spent 70% of the season closed due to that. And Temescal and Anza were both open for the entirety of the swim season and had no danger advisories, which is huge. And then you can see Del Valle had a couple danger days, but for the most part was in caution. Anya will actually be presenting later about the mitigation that she used last year that may have contributed to why Anza and Temescal were not closed at all. You can go to the next slide. Okay, so these are two sites, Contra Loma and Big Break. They are not swim beaches, but they are water bodies that we routinely monitor. And the background photo is actually from Contra Loma in July during a particularly intense algae bloom. As you can see from the graphs, Contra Loma spent almost half of the year with a danger advisory, and Big Break spent most of the year with no advisories. So that was nice. Next slide. This final slide is from last year at the beginning of August when my colleague Anya noticed microcystin cyanobacteria at the Martinez Marina, which is near the 680 Benicia Martinez Bridge. Upon further inspection, it was also located along the shoreline that includes our Radke Martinez Regional Shoreline Park. This HABs bloom is the first time the park district has placed advisories at this location, and strip testing revealed that microcystin levels were greater than 20 ppb for the almost 3 weeks that this bloom was active, which was particularly interesting since this is brackish water. So we were learning about how microcystins toxins persist in a different environment. Cool things that came out of this incident is that it gave us the opportunity to communicate our HABs advisory system to a park that was unfamiliar with those protocols since we had never posted there before. And we were also able to provide guidance to the city of Martinez on how to communicate with the public about this bloom. But yeah, that's all I have for the update. Feel free to reach out to me, Hal, or Sabrina if you have any questions about what's going on with our parks or the updates we do on HABs.
All right. Thank you so much for your update. Also, just wanted to throw in there before we move on to Region 3, I saw in the chat some people are asking about Brianna Cicada's monitoring, and she's going to be presenting today after Region 5 or in Region 5 section. So we will get an update. I know she's kind of got a statewide program, so I didn't know where to put her in, but I threw it in with Region 5 for this time. So we will be getting an update in a bit. But for now, let's go on to Region 3. Melissa, are you here?
I am. Can you hear me okay?
Yes, I can. Thank you.
I think I put a slide deck in there too, if you want to pull that up.
Do you see it?
I do. I recycled this from a different meeting, so also there'll be some animations, so I'll let you know.
No worries.
So yeah, my name is Melissa Doherty. I'm the regional monitoring coordinator for the Central Coast Water Board. We actually don't have a regional HABs coordinator position at this office, so I act in that role. I co-manage our Central Coast Ambient Monitoring Program, which is a partner monitoring program known as CCAMP. That's why it's a CCAMP on this slide. So because we don't have dedicated resources for HABs monitoring in our region, we leverage our CCAMP partner service water monitoring program to do this work with myself, and occasionally we borrow Region 3 staff. So next slide. So generally speaking, these are the three major components of our program that best describe the HABs monitoring efforts that occur in our region. So typically we participate in the holiday assessments. We work with the state water board leads to do some response monitoring or bloom follow-up monitoring when we get reports or if we see some imagery on the satellite map that indicates a bloom might be happening. And when we have additional resources, we do participate in special studies. Next slide. So this, again, is kind of showing the way we prioritize our monitoring in our region. So typically there are these two categories, or prioritized monitoring and our special studies monitoring. So for prioritized monitoring, we participate in the statewide holiday assessment efforts, trying to target major holiday weekends at some of our high use recreation water bodies. And we'll also conduct investigative or follow-up monitoring in response to notifications from the online bloom reports or the satellite imagery, as I mentioned, or working with state water board staff when there's reports from the public coming in about a potential bloom. And so again, if there's resources available, we also will participate in some special studies. And luckily for us this year, we're actually going to be using the new FICO probe to do some more extensive monitoring at one of our lakes that have typically experienced HABs year round and many years in a row. So next slide. So this is a map just showing the locations of the 6 water bodies that we typically sample during our pre-holiday assessment. So they're listed there on the left, and then you can see where they are on the right. And when we go to these locations, we make sure that we're sharing the information with the FHAB program so that the results of our events can be posted on the online map. But we also work with our local water body managers to let them know the results of the sampling efforts. And if there's an advisory, we'll let them know which one to post. So you'll see that. And then if you click it, there's a animation on this slide. So again, this is the map, which shows the colored dots on this map are representative of what was the end of last year's monitoring effort. So this was the suggested advisories after the end of the pre-Labor Day assessment. If you click one more time, it just kind of shows what those mean. We didn't have any danger suggested advisories this year, which was really great. We've had those in the past. And so the highest advisory level we experienced in the 2025 season was caution. And then we had some general awareness based on some visual observations at some of our locations. So, next slide. This is basically summarizing the same information. So the lakes on the left there had no advisories during the different parts of the holiday assessment. We did have general awareness at Lake San Antonio. That's actually the lake we're going to be deploying the FICO probe. They do tend to have HAB conditions pretty regularly, and not always do they have toxins, so that they're not always detected there, but we do often see cyano species that are toxin producers. So that's the lake we're going to be targeting this year. And again, the highest caution or the highest advisory level we experienced in our region at these water bodies this year was caution for Oso Flaco and Lopez Lake. Next slide. Again, this is kind of a summary of every pre-holiday event for each of those 6 water bodies. And so again, we were pretty lucky this year. We didn't have any really high toxin detections at some of these water bodies where we've experienced those in the past. So caution was the highest advisory level. And Oso Flaco Lake is another lake that tends to have bloom conditions, if not visual indicators year round. So they had caution posted, and they tend to have caution posted year round at that location. So, next slide. So just some photos from that last sampling event. So you can see on that bottom right corner, those are photos from Oso Flaco Lake and Oso Flaco Creek. So that lake tends to be very green year round. So you can see that kind of pea soup color. So even though there's not necessarily toxins detected, we do tend to have a lot of high cell counts at that location. So next slide, I think just wants to, summarize some of the partners we work with. And again, this is done, the monitoring is done with our CCHAB field team that go out and collect the samples. And then we just make sure that we're coordinating as much as we can with our local water body managers to share that information. We share it internally with the office. We make sure that it's posted on the online web maps. And we work with our community to share any resources or outreach materials that are provided by the FHAB program, and also offer sampling advice for some of our lakes where they sample outside of that pre-holiday event season. So most of our funded sampling events are because of the efforts that the FHAB program has given to the regions to support some of that pre-holiday assessment monitoring. So I just wanted to thank you guys for all the support you give to the regions that don't have dedicated funds for FHAB or for HABs monitoring. So that's it for Region 3.
Great, thank you. All right, on to Region 4, LA. Emily, are you here?
Yeah, can you hear me okay?
I can.
Cool. So I think there's another slide. Hi, I'm Emily Duncan. I'm swamp coordinator. We don't have a paid position for FHAB, so I cover that too. And I'll be going on maternity leave starting in March, so I'll be working on getting this program covered. So I'll keep you guys posted on who the point person will be. But just an FYI for 2026. So pre-holiday assessment, I realized I didn't put the caution warning information on this table, but basically I know Brienne will talk about Pyramid and Castaic later. Those are our regularly monitored water bodies by DWR, and you can see, like, I just put the range of concentrations. So we did get pretty high microcystin concentrations in Castaic over the spring, summer, fall. It's still being monitored. And then our partner monitoring for the pre-holiday assessment, we've done 4 water bodies the last couple of years. I think I'm going to be trying to increase that number going into 2026. But we collaborate with LA City for Machado, and then LA County Parks for Leg, Pudding Stone, and Santa Fe Dam. And so you can see Santa Fe Dam didn't have any cautions or warnings, any detections, microcystin, or any toxins. And then we did have some lower levels at Machado. That water body also has a TMDL, so the city has various projects going on for that water body. And then Lake, we did see some microcystin. So we worked with them to make sure that they had proper signage. And that was something that I did when I sent everybody their kits, was also send them a bunch of different informational and then caution, warning, danger signs. So yeah, that's the pre-holiday assessment, gearing up for sending out those sample boxes again this year and hoping to get these same lakes and a couple more. Next slide. Yeah, thanks. So there were about 4 incident responses over last year. I think the most notable one was probably Venice Canals, thanks to Carly and Melissa for helping our regional board handle some of that. LA County was technically the lead, but there were high concentrations of cylindrospermopsin and anatoxin, well, mostly cylindrospermopsin in the scum samples and benthic mats that folks sampled from my office. And that got a lot of attention because of dog deaths. Still unclear to me whether it was I think it was, I don't know. I never heard a conclusion of that situation, but a bunch of dogs died and it got a lot of attention in Venice Canals. Elizabeth Lake is a tricky one. Forest Service has like a boat launch there, so I was able to send them some equipment. I don't believe they were able to follow through with sampling. That's one that we definitely keep an eye on because those are lakes that are seasonal depending on rain. They can dry up occasionally. So they're kind of an interesting Lake Hughes and Lake Elizabeth. They're rift, like tectonic rift lakes. And then I did get a call from a private citizen about the ditch that runs through their neighborhood in their backyard. Was able to sample that for them and no toxins detected, just some algal mats that they were concerned about. And then there was some sampling at Big Rock Preserve on Ventura River, which staff in my office also worked with Carly on because they have the Surfrider group there has like an active monitoring program in that watershed. So yeah, I think generally not too crazy over 2025 in terms of our blooms or reported blooms, but Pyramid and Castaic were active, specifically Castaic. So yeah, that's what I have for 2025.
Great. Thank you, Emily, and congratulations.
Thanks.
All right. Anyone else for LA before I move on? Okay. Then we will move on to Region 5. Dana, we'll have you go first. We've got quite a few folks for Region 5 today, so.
Good morning. You can go ahead to the next slide. I'm Dana Schultz, the FHAB program coordinator for the Central Valley Water Board, or Region 5. And you can see our area in the map there is the light blue area that spans through the Central Valley. So looking at our 2025 HAB event response kind of year in review, we received 53 bloom reports from late February through the end of November 2025. The large majority of those, or 89%, were received via the online water board supported bloom report form. They came from 30 unique water bodies across 14 counties. And then within those bloom reports, the HAB illness work group investigated 23 of them from our region. And per their last email update, it looks like 4 reports were determined to be HAB related and submitted to the OHAB's database. And these included 3 fish events and 1 dog event. You can go ahead to the next slide. And our office participated in the FHAB program pre-holiday testing. I shared this at the last CCHAB meeting, so I'll keep this pretty brief, but we have a staff member down in our Fresno office, Akira, who goes out and samples Hensley and Eastman Lakes, and they both had a couple advisory postings this summer. Thanks to Big Valley Band of Pomo Indians for participating as well. We support a couple of those sites during those time periods, but I point you to their full data set to take a look at that data and advisories. We also supported a collaborator with McCord Environmental who tested Pine Mountain Lake and the nearby Bass Pond this past summer, and there were no advisories there. And thanks to the other groups who also coordinate their testing to fall in these windows and share results with us. You can go ahead to the next slide. So moving into partner monitoring, our office participated in a couple partner monitoring programs this past summer. So first, work with Restore the Delta. Multiple water board agencies have been working with Restore the Delta since the summer of 2022 to sample for HABs near Stockton. So this past summer, their field crews went out to sample 7 sites. They're going out weekly to deploy a sonde and do visual HAB assessments and screen for microcystins with the rapid test strips. And then this past summer, there were 6 sample events where they did water grabs and sent it to Ben Genetics for microcystin ELISA testing. And this past summer, that was funded by the Division of Water Rights. And looking at those results, out of 42 samples sent in, there were 5 microcystin detects. The concentrations ranged from 0.47 to 118 micrograms per liter. And that high value was one of 2 scum samples sent into the lab this past summer. Next slide, please. And then the other big HAB partner monitoring effort our office is involved with is the Kaweah River HAB monitoring. And we've partnered with Tulare County Environmental Health for the past 2 summers. They've collected water grabs, deployed spat, and collected benthic mats if present. So we sent in a couple benthic mat samples, potential benthic mat samples this past summer, and the lab let us know they were predominantly green algae, diatoms, detritus. So over the past 2 summers, the county staff that were going out didn't see any clear areas that were dominated by benthic mats. There may be some cyanobacteria kind of in the system, but no clear areas of cyanobacteria benthic mats. There were some cyanotoxin detects in the spat, but they were all less than that kind of 100 nanograms per gram threshold that's been put out with CCHAB guidance. And there were some rare, but there were a couple cyanotoxin detects in the water grabs, but they were all low concentrations, and it was pretty rare. I think only there was 1 antitoxin detect, which led to a caution advisory, just because detection in CCHAB guidance is caution, but I believe that was the only water grab that led to a posting. And then just some context here, this partnership came about after there were a number of bloom reports in 2022 in this area. And 2022 was a really dry year with not a lot of flow, coming kind of on the back of a few dry years before that. And then 2023 was an extremely wet year, and 2024 and 2025 are also wet years. So what we sampled in 2024 and 25 was kind of a different environmental context than some of the previous years. And that's my update.
Great. Thank you, Dana. Next, we have Brianne Sakata.
Hello. Thanks for having me to give an update. So I'm Brianne Sakata. I lead DWR State Water Project's cyanotoxin monitoring program. And this is showing a lovely photo of the current conditions at Castaic Lake. You can go to the next slide. So just an overview of our monitoring program. As you can see, we cover multiple water board regions. We monitor both water quality and recreation sites in the SWP. Our routine monitoring program involves twice monthly sample collection at our water quality sites from April through October. And then we sample recreation areas at our reservoirs weekly from Memorial Day to Labor Day. And we do sample outside of our routine season in response to bloom observations, or sometimes blooms will extend beyond our normal monitoring season, like Castaic, and we continue to monitor blooms until they're gone. So our water grab samples, they're screened for potentially toxigenic cyanobacteria by microscopy, and that's followed by toxin analysis if it's recommended based on the microscopy results. Next slide. A summary of our advisories during 2025. We had 11 water bodies posted with caution. Lake Davis, Antelope Lake, and Lake Oroville were posted just due to visual indicators. There weren't actually toxin detections at those lakes. Lake Del Valle is a state water project lake, but the recreation is managed by East Bay Park, so that's part of their program, so that's their posting. So caution was also posted at Los Banos Creek Reservoir, O'Neill Forebay, San Luis Reservoir, Quail Lake, Pyramid Lake, Castaic Lake, and Silverwood Lake. And then we had warning advisories at San Luis, Pyramid, Castaic, and Silverwood in 2025. And then danger at Lake Del Valle, Castaic Lake, and Silverwood Lake. Next slide. So these are our 4 reservoirs that had the highest microcystin concentrations in 2025. San Luis Reservoir ranged from 0.37 to 7.5 micrograms per liter. Pyramid ranged from 0.72 to 12.35. Castaic ranged from 0.47 to 94, and I have an asterisk there because this bloom is still ongoing, and a sample we collected last week actually had 3,150 micrograms per liter of microcystin, and that's the highest number I've seen in our monitoring. And then Silverwood, the microcystin concentrations range from 0.35 to 32.7, and at all 4 of these lakes, the most common cyanobacteria attacks were Microcystis, Dolichospermum, and Aphanizomenon. The bloom at Pyramid Lake was dominated by Dolichospermum and Aphanizomenon, but the other 3 were dominated by, or are dominated by Microcystis. And that's my last slide. Thank you.
Great. Thank you so much. Okay, and now we have Spencer from Restore the Delta is going to give a little update on his program.
Yeah, thanks for passing it over. Hi, hey everyone. My name is Spencer Fern, and I'm the science program manager with Restore the Delta. Going to give an update on what our harmful algal bloom monitoring looked like. But if we could go to the next slide, I'll talk about what Restore the Delta does. So Restore the Delta has worked since 2006 in areas of public education and outreach, and has grown over 75,000 followers throughout California. Our goal and vision is we envision the Sacramento-San Joaquin Delta as a place where vibrant local economy, tourism, recreation, farming, wildlife, and fisheries thrive as a result of collaborative efforts to protect our waterways, and monitoring for harmful algal blooms ties into the water quality aspect of that. And our goal is to connect communities to our regional rivers and empower communities to become the guardians of the estuary. And ideally build the next generation of water leaders, which part of the program with the interns I work with is trying to build a scientist to agency pipeline with college age students who work in our program. Next slide, please. So to kind of just highlight what we were experiencing in Stockton in 2025, Dana did a good job of setting the runway for the testing we've done and how we've been working on this since 2022. But some of the samples we've done through 7 sites, and the areas that we did sample, there were 3 sites that experienced bloom activity in 2025. One of them is pictured to the right, which is at the Lewis Park boat launch. And then there was also another one at the Buckley Cove boat launch, as well as off of the levee near Windmill Cove, off of the Windmill Cove Road. Each of the sites experienced the similar chopped grass appearance of bloom activity through these, and we were using gold standard diagnostics, Abraxas test strips, and that maxed out at 10 parts per billion. But each of these, or specifically the Lewis Park boat launch, maxed out, and as it was mentioned on the in Dana's slide, we did send samples off to Ben Genetics, and for this site specifically in early August, it reached 118.13 micrograms per liter at Lewis Park. Something to highlight is like what I had mentioned, like we have our interns come out and test, and a lot of them were able to get experience doing this work and kind of get the exposure to harmful algal bloom monitoring, and this was like the first time we'd experienced blooms in any of the sites that we'd sampled for, I want to say, since 2022, aside from one or two off-ball results in previous years when there weren't visual blooms, but these are the first times we've seen visual blooms since 2022. And that's kind of like the rough update of what happened in 2025. But this boat launch at Lewis Park, I should provide some context, was closed off to the public for a number of years until they had finished a gate project over in that area of the waterway. So that would, and now that they've finished that project, it was open to the public. And while I was out there grabbing samples, I did see people jumping into this water, launching kayaks, fishing off the banks, getting it up in their boats. So highly recreatable area, something that we're trying to keep an eye on for community perspective and keeping people updated on that. Next slide, please. So the next steps for community harmful algal bloom monitoring is trying to continue to be the voice between agencies and the public in the Delta. I mentioned in the just right now, I just mentioned how we're trying to keep the community safe. Part of that is making sure that when we're collecting samples, signage gets posted soon in ways that people can see them. There's enough information out there for people to readily access because a lot of people around the Delta, at least in the Stockton area, aren't aware of harmful algal blooms, especially with the amount of times I'm out in the field and people are asking us what we're doing. I mean, you can see the picture of one of my interns in the picture there. We're wearing bright vests and we're carrying long, like, poles to pick up water. It definitely looks pretty serious sometimes, and people have questions about that. Another thing we're working on is we're in a partnership with DWR with Ellen Preece, as well as I think it's a few other programs, but we're working at sampling areas around the Delta or around the Deepwater Shipping Channel and then dredging it in a few years and then seeing if there's an improvement to look at if there are if we remove the sediment that holds microcystins or other HABs over the summertime or the winter time frames, that they might not show up the next summer. So that's a project we're working on in the future, and hopefully there are some things to be learned from that going on to maybe act as a mitigation in the future for other HABs in other parts of the Delta. And then we're also working on possible partnerships within the community with local universities and other organizations to possibly expand monitoring and maybe run different lab procedures. But as of this moment, those are just conversations. But those are kind of the processes of what Restore the Delta is working on. And we'll soon be hiring a new group of interns coming up here pretty soon to start testing early May and into September coming this year. So I think that's it for my slides. I think the next slide's just like where you can keep up with us, or if not, that's it for me.
Oh, that's it. Okay, great. Thanks, Spencer. Yeah, we look forward to hearing about your results at some point from the dredging study.
Thanks, Fred.
Sarah, did you want to talk about your monitoring program at all?
Yeah, absolutely. I'm going to share my screen.
Oh, great. Let me stop sharing a second. Or can you just take it over? Oh, here it is. Sorry.
Hi. How are you feeling?
Hey everybody, I'm Sarah Ryan with the Big Valley Band of Pomo Indians. We've been doing the cyanotoxin monitoring on Clear Lake since 2014. We started the program with Elam Indian Colony, which is another shoreline tribe on Clear Lake, and since 2020, they have not been participating, so the program is something that we're running now since 2020. So I am showing during the summertime, we go out twice a month to about 23 locations. During the winter, we're just going out once a month to about 10 locations on the lake. The majority of these are shoreline sites, although we do partner with Lake County, who goes out to the center of each arm of Clear Lake to do some additional monitoring, and they bring back water samples to us, which we do microscopy for and then send off for toxin analysis based on the cyanobacteria that we're seeing dominant and secondary in our microscopy. So it's been a really interesting year. For those who've seen reports on Clear Lake over the years, this is like 2016 in terms of cyanobacteria and cyanotoxin levels. So we have this caution, warning, and danger is based on the thresholds that have been identified through OEHA's 6 cyanotoxin study, and there are adopted by CCHAB in the signage. So the caution is anything that is between 0.8 micrograms per liter and 6 micrograms per liter for microcystin. We do analysis of other toxins, anatoxin in particular, when we see an anatoxin, a potential anatoxin producing cyanobacteria dominating, but we rarely see it. We've also done qPCR, sent off for qPCR analysis through Ben Genetics in Sacramento, and I think maybe just a couple of times we might have seen some saxitoxin producing genes, but not seen saxitoxin in the lake. In any case, throughout the whole year at each of our locations, that's the column B, the site ID, we did not have a single location, if you look at it from the whole year, that had anywhere near 50% of the time being impaired in some fashion. But if you look at it just from the summertime analysis, which is we're basically saying June through October, early October, there were a couple of sites that had about 50% of the time in at caution, warning, or danger, but it was just not that year for it. We had a couple of sites that had danger levels of cyanotoxins. Matter of fact, the highest toxin level that we had was 1,376, and it was at the Sulphur Bank Mercury mine. One of the interesting things was that we saw a dominance of Seneca cystis for the cyanobacteria in many of the sites starting in June. Starting in July. So we keep, we have like tracking things that we do so that we can communicate the results. So yes, so Seneca cystis, even though it is a microcystin producer, it doesn't appear to be. We didn't see much microcystin in locations that had a Seneca cystis dominance. The other thing that happened, so it was an interesting year in terms of that, and also just being able to track Seneca cystis and seeing what I did in this lake. The other thing that happened that was interesting this year was there was a fish kill that happened in, and I just want to pull that report up. There was a fish kill that happened in September that was very it killed a number of fish. It was something that many people were concerned about, potentially millions of fish of many different species. And so we started to look at what was going on at the time of it, which was early September. And it was there were fish that were noted throughout the whole lake. So right around when the fish kill happened, we were seeing a dominance of Seneca cystis, and here's 3 different locations, and that's what the Seneca cystis looked like in the site. And then the following, let's say that was 2 weeks later. So this was kind of the end of the fish kill, the same locations. There was still Seneca cystis dominance. The toxin levels, though, this is what it looks like under the microscope. These things right here, they look like double cells. The toxin levels were not that high. So if there was a drop off of a die off of the bloom, it was not very obvious. This was the satellite imagery during the fish kill. Is not that we've certainly seen a lot higher phycocyanin in the lake during blooms. So it's just Seneca cystis is just an interesting cyanobacteria. I want to also go over some results that we've finalized for cyanotoxins in fish. This is a reminder, we've already reported on this in previous presentations, but we were looking at a number of fish that were that we had from 8 years from 2010 to 2018. And this was our first study looking at microcystin in the tissue. And just wanted to point out that the fish that had an average higher than the 10 nanograms per gram, which is a suggested action level for for sports fish consumption, which is 1 fish meal a week. And we looked at it from a spatial, temporal, and species specific review of microcystin in these fish. So here's the results from the second study that we did that had fish from 2017 to 2023. We looked at 79 different fish, and we were only looking at the tissue. And this was we were seeing lower numbers in terms of the average of microcystin in both where they were caught, when they were caught, including the year and the season. The highest microcystin level was in the Sacramento blackfish, and it was 7.19 micrograms per liter, which is definitely lower than the highest levels that we saw with the first study that we did. So we'll continue to send off fish that we collect during over our different events for microcystin. We'll continue to report on this. This still, there's the suggested action level for 1 fish meal a week is at 10 nanograms per gram, but there is subsistence consumption that happens on Clear Lake. So we're very interested in having a number that could be used as a threshold. We're recommending a 2 nanograms per gram. We're hoping to work with OEHA on that. But with the fish meals a week, if we have people who are eating multiple fish a week from this lake because of tribal consumption rates, and we want to have that threshold. And I think that's all I wanted to share. Thank you so much.
Great, thank you, Sarah. Okay. Now we're on to Region 6. I think Marissa or Carly, were you gonna fill in for Sabrina here?
Yeah, hey Karen. As SWRCB and I'll be supporting Regional Board 6 or the Huntington region. This material was prepared by Sabrina Rice, the FAB program coordinator for that region. And I know the map already went away, just to orient folks. Now we're moving to the Huntington region, which is a higher elevation. It's a very long, narrow regional board spanning from the Oregon border down, through most of a good portion of San Bernardino County. Thank you. There in that yellow colored. Thanks. All right. They're presenting the information that was collected from their extensive partner monitoring program and the holiday assessments that are done in the Huntington region to surveil for potential harmful algal blooms in many of the lakes and reservoirs in these higher elevation areas, particularly lots of parks, lots of boat launches. There's lots of recreation happening throughout the warmer months. Next slide. All right, so this is some photos that capture some of the unique observations of the cyanobacteria blooms that were occurring across that region. Just noting, different colorations, different kind of surface accumulations. It just really helps with their public communication. Also, you're not always looking for the bright blue-green. Sometimes it's different shades. Sometimes it's more subsurface than that film or that scum layer. Next slide. All right, so this is much of their data set that they pulled together to answer some of the questions for these year-end summaries. So this is showing the data that's been collected since the onset of HAB monitoring in the region. So let's look at 2025 for that summary in the far right. So 131 sites were sampled in 2025 across that region by the Regional Water Board and their extensive partner monitoring entities that participate. that monitoring resulted in 15 water bodies with caution postings. That's the lowest CCHAB advisory, recommending dogs to stay out of the water and children as well. And then the other advisories that were recommended from the monitoring were at 3 different water bodies at the danger advisory. So that's the highest, including no swimming, but also staying out of the water entirely, even from water wreck from boats. So that advisory was posted at Bridgeport Reservoir, Crowley Lake, and Silverwood. Silverwood also was brought up from Rihanna Sakata, it's part of the State Water Project as well. And also they wanted to highlight the illnesses reported. So these are suspected related illnesses that are just preliminarily reported from either the public or from other partner entities. And that's the preliminary, and then the final confirmation from the illness work group is presented below. So you see it's quite the difference. There's suspected illnesses for fish, dogs, and human exposure in that region. Next slide. All right. So the types of monitoring that was conducted throughout the Lodgen region included, response to new blooms from the online bloom reporting that comes from the website, and also monitoring of recurring blooms. That's something that we see in many regions. There's a lot of recurring blooms happening at our lakes and reservoirs. They do incident response, and also I wanted to highlight their extensive partner monitoring folks. So of note, there was routine monitoring, not just from the incident response. So the routine monitoring was conducted by Inyo County Environmental Health at Diaz Lake, Mill Pond, Pleasant Valley Reservoir, and Owens River. And then Los Angeles Department of Water and Power also participated doing some routine monitoring at Crowley Lake, Pleasant Valley, and Tillamook reservoirs. And also there's extensive pre-holiday assessments. Next slide. All right, and so this is summary of what was done during those pre-holiday assessments. They do focus most of their assessments after, starting 4th of July and then through the post-holiday season around October, November, the last sampling. There's upwards of 25 water bodies that were sampled for the holiday assessments. And then, unfortunately, the color coding didn't come through on the table, but the lower rows show like the different advisories that were resulted from the the holiday assessments. And also there was monitoring for benthics blooms as well. So there was some algae alerts or not just planktonic monitoring. Next slide. All right, so this is really great table to pull together all the information on that partner monitoring, really highlighting the wide range of partners, Watershed Council, Fish and Wildlife, the department, there's utility districts, homeowners associations, health departments, as we had spoken about, park districts, many different tribes have participated in the past and even participate still this past year. And just really want to say thank you, extend Sabrina's big thank you and her team's appreciation of all the different partners that monitored across the region. Its region is very long and narrow. It would be impossible for the Regional Water Board to be able to cover this amount of lakes. And also, I think this spans 8 different counties, this partnership program. So thank you, everyone. I think that's the last slide. Go ahead.
Yeah, thank you so much.
Okay. All right. Thank you.
On to Region 7. Jeff, are you here?
I am.
Awesome.
Okay. Start off with Palo Verde Lagoon. It's a lagoon that's located on the lower Colorado. It's like a small diversion of a drain that passes through a trailer park. Back in September, we had a lot of heavy rains and it backed up the lagoon. Several of the banks got damaged in the area, and that allowed the flows to carry in a lot of trash and nutrients and sediments and things. It even caused a huge pileup of debris that ultimately plugged the outlet. And of course, that resulted in a fish kill. I did go on scene. The smell was pretty bad. I did find cyanobacteria and toxins, but they were at really low levels, below the threshold. So essentially, the lagoon had become so stagnant from the backup that the oxygen got depleted and just killed off the fish. The water district kind of deflected the responsibility for the cleanup of the fish, so the residents had to take it into their own hands and clean it up themselves. Although the water district did apply to FEMA, so they were able to fix the outlet, get it unplugged, and they also fixed the damaged banks. Next slide. Weast Lake has been experiencing chronic blooms for years. It's a county park where there's swimming and fishing, but lately it's been closed to recreation because of the HABs. We mainly see Raphidiopsis whenever there's a bloom, although we do see microcystin on occasion as well. Now, I've proposed an assessment there to kind of determine what's happening within the lake and the sediments. But the problem is that even if we come up with a solution, the county hasn't really committed to following through with any of our findings to implement any kind of solution. So it's hard to justify that work, especially now since we don't really have any funding. So maybe that's something we can revisit this year. Let's see. Next slide. And that takes us to Salton Sea. Salton Sea has been experiencing HABs for a number of years, since it's dominated by ag runoff and sewage. And so it gets a steady influx of nutrients. Most of the shoreline, it has a perpetual benthic mat that contains over a dozen varieties of cyanobacteria. It's pretty common to go out there and sample and find multiple varieties in a single sample. Right now, we have a permanent caution level advisory for benthic HABs at Salton Sea, and that's been in place for, I think, 2 years now, maybe. And in 25, we had a guy who was wearing jeans sitting on a sofa in the shallow part of the sea, because why wouldn't you sit in a sofa surrounded by benthic algal mats? And so, long story short, he developed a pretty nasty rash and some sores on his legs that did require medical attention. But we never did determine that HABs were the cause, since he had also visited one of the hot springs in the area. So we couldn't really determine the cause. Next slide. And finally, we have Lake Havasu. We had multiple blooms reported in Havasu in 25. Most of these occurred on the Arizona side of the lake, though, and that's handled by the Arizona Department of Environmental Quality, who also happens to be one of our monitoring partners. We're just nearing the end of our 12-month HAB study there with Arizona and Arizona State and EPA, BLM. It's a multi-agency collaboration. And we've got, I think, 4 months left until we wrap up that study. So that's all I have. Thank you.
Thanks. All right, Santa Ana, Region 8, Kirk.
Do you have the slides you can share?
Yeah.
Okay, there we go. Great. Can you hear me?
Yes, I can.
All right, awesome. All right, Kirk Larkin, the Harmful Algal Bloom Coordinator for the Santa Ana Regional Water Quality Control Board. Next slide, please. So in 2025, we did our pre and post holiday assessments at 5 different lakes, Big Bear, Lake Elsinore, Lake Hemet, as well as Mill Creek and Lytle Creek. And moving forward for 2026, probably similar locations. We're still identifying which locations we may monitor for, but it might be very similar. Next slide, please. So here's a summary of our data for 2026. As mentioned, these particular water bodies were sampled prior to Memorial Day, 4th of July, and Labor Day. We focused on collecting extra samples from the lakes, Lake Hemet, Lake Elsinore, and Big Bear Lake for the major holidays. But then we also had some information where there's caution advisories posted for Lake Hemet and for 4th of July, elevated to danger for Labor Day. But based on that information, we did post-holiday sampling as well. That information came back as danger advisory recommendations as well. Lake Elsinore, they are doing some mitigation there, definitely improvements in water quality based on historical data. There was a discrepancy for the Labor Day samples. Water board staff are collecting samples for these events, but the city of Lake Elsinore also collects samples and are sharing data with us that we help post on the advisory notifications as well. But for the Labor Day sample, they collected information where they had ELISA testing as well as LC-MS testing. There were some discrepancies in the data. We erred on caution to actually post an advisory notification because the LC-MS data were showing that they had exceedances for microcystins. Interesting enough, the ELISA testing were below the caution threshold, but again, we elected to post a caution for that particular event. City of Big Bear Lake, Big Bear Lake, throughout the season, they had no advisories, but based on satellite imagery, we noticed that they were having a bloom later on post-season. So we did collect samples on the lake, which came back at a warning advisory. For Big Bear, we're actually sampling at 2 locations. For the advisory, we sampled along the North Shore, which came back with fairly high microcystin levels. And then we also collected a sample along the west end of the lake at Stanfield Cutoff, which was still detect microcystins, but ultimately went with a warning advisory for the lake. We did not see any benthic blooms or significant planktonic type blooms for Mill Creek or Lido Creek, so there's no advisories posted for them. Next slide, please. I'm not seeing the slide advance. Am I frozen on my end?
Bloom and illness reports, is that the one you wanted?
Oh yeah, okay, here it is. Sorry for the delay. So here's a summary of the bloom and illness reports that we had for the Santa Ana River region. As you notice, there's a handful or half a dozen different reports that came through. 3 of those actually were identified as HAB related. Lake Paris, it wasn't actually a bloom illness report, but just the monitoring data that DWR shared. They had a slight elevation for microcystins in July, so that was reported. It was HAB related. Canyon Lake, they had a report of a fish death that was attributed to a golden algae bloom that had occurred. And then we received a notification for a small park down in Orange County where there was an observed fish death. And so we had a HAB advisory for that one as well. And that's all I have for Region 8.
Thank you so much, Kirk.
Yep.
All right. Last but not least, we've got Carrie with San Diego.
All right. Are you sharing my slides for me? Oh,
Yes.
perfect. All right. Hi, I'm Carrie Kowalski, and we don't have a dedicated position for FHABs, but I'm the coordinator for Region 9. And unfortunately, I just lost my partner who helped me a lot, Debbie Woodward, but she's retired, so we're happy for her. So next slide. So over 2025, our main focuses were the incident response and some satellite notifications. With that, we had to investigate 10 separate water bodies. We did our pre-holiday assessments, which were 5 sites that were funded by State Board. And with those, we tended to see the blooms later in the year. So 2 of our sites out of the 5 required postings after our Labor Day sampling. And then finally, we did a small special study with the FICO probe that was purchased by State Board. And I'll talk a little bit more about that later. I have a presentation later today focused on Lake Henshaw, which was where we did the FICO probe work. Next slide. So I just wanted to go over just a few of the notable blooms of this past year. One was at Whalen Lake, and that was through an incident response in the spring. And the lake was experiencing a microcystis bloom, and the microcystins were at the caution level at that time. And this lake is important because it is a bird sanctuary, and it also receives recycled water from the San Luis Rey wastewater treatment plant. So the landowner and they work with the wastewater treatment plant and let them know what the conditions that they're seeing, and so they can kind of fluctuate how much water is released to the lake. And they also have installed an aeration system to deal with the blooms. So by October, there were still microcystis present, but the levels of the toxins were below a posting level. But it's nice to have the manager there on site at all times, so he lets us know what he's seeing. And then also we're concerned because he does have a dog that frequents the lake, so give him information about that. Another bloom that we've seen since the fall, it's still in bloom, is at Santee Lakes. And this one has more of a variety of cyanobacteria causing the bloom, and the microcystins are at a caution level at this time still. Next slide. Cedar Creek Falls, this is a high recreational use area that was part of our pre-holiday assessments. And unfortunately, during the last Labor Day sampling, there was microcoleus, and microcystins were at a caution level. So we continued monitoring with especially after the rains, we were hoping that that would flush out the system. But again, in November, it was still staying at a caution level. However, it shifted. The main cyanobacteria species causing the issues was microcystis. And then again, after we had rains in December, we went toward the end of the month. Very hopeful that the conditions changed for the better, but however, it's more microcystis, and the microcystins now are at a warning level, so that's to be continued, and we're working with the Forest Service with that and sharing our data with them. Finally, the other bloom I'll mention from our region is at Diamond Valley Lake, and that one started in the springtime, and it was at a caution level. It's Metropolitan Water District who takes care of the testing and shares information with us. So that was microcystis at the time. And then they were bloom free for most of the fall. In December, they're back at a caution level. And that bloom is dominated by Limnoraph. I'm not sure how to say it exactly, Limnorapsis species, but that was recently separated from the Lingbia group. And while that species isn't known for producing toxins, it's typically occurs with microcystis, Dolichospermum, and other toxin producers. So that's why we decided to keep it at a caution level for now. Next slide. Just wanted to mention some of the collaborators that we worked with throughout this season. And Mission Trails Regional Park, we had a few sites there. They were great about sharing information with us. San Diego River Park Foundation also helped with posting signs and giving us some sites that they would like for us to sample. Next slide. And then finally, our FICROprobe special study, which I'll talk about a little later, happened at Lake Henshaw, and we did monthly outings from May through October. So it was just once a month, but you can see the device that we had there on the left, the big FICROprobe. And then on the right, some of the Vista Irrigation District staff that were graciously let us go out on the boat with them. And I think that might be it now.
All right. Well, thank you so much to all of our coordinators and partners who presented today. You guys are doing incredible work across the state. We will now adjourn for 10 minutes to go do some bio breaks and grab some coffee stretch, and we will be back at 10:40. All right, everybody. We're gonna resume our meeting with an exciting research presentation. We have Anya Bray from East Bay Regional Parks District here today, and she's going to tell us about Happier Lakes, I love the title, and she's really going to tell us about the nutrient reduction in Lake Anza and Lake Temescal. So over to you, Anya.
Thank you. Thank you, Karen. Sorry, my camera is not where I'm looking. I think that's why I'm going to turn it off during the presentation. So thank you, everyone, for being here today and for being interested in my talk. I'm going to be presenting on the 2025 lake management activities at Lake Anza and Lake Temescal, and I'm an ecological services coordinator at the East Bay Regional Park District. So I wanted to start out with a slide that shows a bunch of black and white photos of the two lakes, Lake Anza and Lake Temescal, that I'm talking about today. And I wanted to do this just to say that these lakes are not natural lakes, but man-made reservoirs. And there was a time before them, like if you look at the photo on the left, that's Wildcat Creek Waterfall in 1920. That was before Lake Anza was built in 1938, and the photos of the right side are Lake Temescal in 1913, where it actually had an extra arm that it doesn't have anymore now, and then another photo from 1940, and that reservoir was built in 1868. And the reason I wanted to show these is because aging reservoirs come with a bunch of problems. And Lake Anza and Lake Temescal, they're small lakes. They're only about 10 acres big. Lake Temescal is very shallow. The deepest spot is about 16.5 feet. Lake Anza is about 45 foot deep. So Lake Anza is about double the volume that Lake Temescal, but they all have these issues that I will be describing here. So one of the issues is loss of depth through sediment infill, sedimentation and infill. The other one is increased nutrient level through external and internal loading, like runoff from the watershed, and then internal loading through sediment. So these lakes are eutrophic or even hypereutrophic. We have low dissolved oxygen level at the bottom of the lakes. In Anza, we installed a hypolimnetic oxygenation system in 2020 that improved the situation, but is not perfect and needs some improvement. And Lake Temescal is pretty bad, as what I will show you later. But yeah, low dissolved oxygen levels at the bottom of the lake due to decaying biomass. Harmful algal blooms, as you can see in the lower left photo at Lake Anza. And then, of course, these aging reservoirs come with aquatic weeds that grow in the lake or along the shore. 2024 was a pretty bad year at the East Bay Regional Park District's reservoirs. Lake Anza was the winner out of the swim season that is 213 days long. The lake was only open 77 days, which means that it was closed 64% of the time. Lake Temescal was not far behind. It was closed 51% of the time. This graph shows microcystin toxins at Lake Temescal in 2023 and 2024. I would like to add here that the district has adopted a different system, a slightly different system than the CCHAB's recommendations. We do not have a warning sign, so we put a caution level sign up when the trigger is 0.8 micrograms per liter and the danger at a trigger level of 6 micrograms per liter. So if you look at these blue bars, you can see lots of toxins up to 50 micrograms per liter. You might wonder why 50. We have our own ELISA CAS machine in the lab, and we test once a week. We only dilute to up to 50 micrograms per liter. That's why the toxin levels stop there. But either way, you can see lots of toxins in September, in July, and then in 24, then like into July. And mostly microcystis is responsible for the microcystin toxins in our lakes. Dissolved oxygen, as I mentioned before, this is Lake Temescal, very low dissolved oxygen in the summer month. If you look at 2024, there was a really extended period of dissolved oxygen that lasted through July, August, and September. We even had some low dissolved oxygen in November. So what were our goals for 2025? We wanted to reduce phosphorus to decrease the occurrence of HABs, but also hopefully to decrease the cyanobacteria toxin levels. So what did we do? At Lake Anza, we were for the first time allowed to do a phosphorus remediation treatment, and we decided to use ACH, aluminum chlorohydrate. And we wanted to build a research study around this treatment to see the success over the 6 months. And so we chose 4 locations in Lake Anza, and we collected samples at 1 meter depth and 0.5 meters above the bottom. And we did this before the treatment, 1 week after, 1, 2, 3, and 6 months after. And then at Lake Temescal, we did the same approach, but instead of just using aluminum chloride, we also added Phoslog in the mix. We did the same study approach as at Lake Anza. And then in addition, we installed a oxygenation saturation technology system to mitigate the low oxygen conditions. So here are just some photos of the treatment at Temescal in case some of you haven't seen what that looks like. There's even an aerial photo from a drone. At Lake Anza, we did the ACH treatment at the end of April to the beginning of May. And at Lake Temescal, we had already a little bit of cyanobacteria in the lake at that time. So we did, on Monday, we did a Green Clean Pro treatment, Green Clean 5.0 treatment. And then on the following 2 days, we did ACH and Phoslog. And we chose to use ACH because it has the same byproduct as alum, aluminum hydroxide. But we used it specifically because it has a much lower impact on pH, which is a really critical factor. ACH has a third of the impact on the pH compared to alum, which is most widely used as a product for water quality enhancement in lakes. So that's the reason why we chose ACH. Then a week later, we installed this OST system at Lake Temescal with the Clarity Resources Group and with Lake Tech together. And now we can look at the results. So let's look first at water clarity. We measure water clarity with a Secchi depth. And for those of you who don't know, it's pretty much a disc that is black and white that you lower into the water column from the boat, and you record that depth where you can't see the disc anymore. So before the treatment, the Secchi depth was 1.6 meters. After the treatment, the depth that I could see the Secchi depth doubled to 3.5, and then 12 days later, we even had 4.6 meters of clarity. This shows Lake Anza, and it was pretty amazing. So I was joking around and calling Lake Anza the new Lake Tahoe, the little Lake Tahoe, just because it was really, really, the difference was mind-blowing. What about the microcystin toxins? This is the same graph that I showed before, but this time it shows 2024 and 2025. As before, there are a lot of toxins in 2024, but if you looked over to 2025, there was a little blip going higher above the caution level just before we did the ACH and Phoslog treatment at Lake Temescal. But after those two management strategies, we had very, very low toxins, most of them below the caution trigger level. We only, for the most part, have caution at Temescal because of benthic cyanobacteria. The last measurement that we did was in December, and it was a 0.96. So really low toxin levels at both lakes, Lake Anza and Lake Temescal. And for the first time in a long time, we had no swim beach closures at both of these lakes, which was an enormous success. What about the total phosphorus levels at the lake? So this graph shows the total phosphorus at the surface in red and the total phosphorus at depth at Lake Temescal. And if you look at 2024, this shows a very typical pattern that we had. So when we had stretches of anoxia in the lake, we would get a lot of phosphorus release out of the sediment, which creates this huge release. And with that, of course, trigger in harmful algal blooms. And if you look over here into May, you can see that the bottom, the hypolimnetic total phosphorus levels already started to increase with the low oxygen levels. But then with the 2 treatments, it dropped back down, and it has been really low ever since until the storm started. And now, of course, we see some runoff come in. But yeah, the phosphorus levels were really low for the remainder of the year. What about the oxygen? I pointed out to you in a previous slide that we had really low dissolved oxygen in 2024. And then in 2025, in May, it started also to become kind of low. But then we installed the oxygenation system, and that worked really well for the remainder of the year. We had no anoxia at the bottom of the lake anymore. Unfortunately, the system is currently out of service, and we're trying to get it repaired. And you probably wonder what this big blip is here in December. So we were wondering ourselves, since the oxygenation system was out. So we went out and we collected some phytoplankton samples, and we had a lot of biomass in it, and it was identified as a diatom Melosyra granulata, and we think that the high oxygen levels, dissolved oxygen levels, came from that diatom bloom. We also took some chlorophyll samples over the duration of the study, and I put a little cheat sheet up there just to show you at what chlorophyll levels we can see what trophic class. So before the treatment down here, it was around 3.8, which is mesotrophic. It means like there's some nutrients in the lake. This is for Lake Anza. And then the chlorophyll level decreased after 1 week of the treatment. That means we probably took some of the biomass out. But then it stayed stable for 2 months, and then we saw a huge increase. And then after 6 months, we saw a decrease into still a eutrophic level of the lake. So chlorophyll is an indicator on how much biomass or phytoplankton it is in the lake, but it doesn't tell you exactly what you have. Is it good or bad phytoplankton? So we decided to also do absolute phytoplankton abundance analysis during this study. And I know this graph is very busy, so let me walk you through it. One thing to consider is that the y-axis is a log scale. So even though these numbers look kind of similar, there is a big difference between those bars. On the x-axis, you see 6 major phytoplankton taxa, with the first one being cyanobacteria. And then the different color shades show you the pretreatment 1 week, 1 month, 2, 3, and 6 months. And what we saw out of these analyses is that for the pretreatment, the 1 week after and the 1 month after, we had this colonial green algae dominate the lake. After 2 months, if you look at the 4th bar, those are these. We had chlorophytes, diatoms, and chrysophytes dominate the lake. And at 3 months, we had this little guy, Malamonas, really dominate. That's where this big spike in chlorophyll came about. And then currently, 6 months after, this diatom here dominates the lake. And I know this is a lot of information, so the takeaway of this slide is that there was a lot of shifting in the plankton community, but the community was not dominated by cyanobacteria through the 6-month study period. And this is even though this was the height of the cyanobacteria growing season. So to summarize all this is we feel like the OST, the oxygenation saturation technology system, created the desired levels of oxygen during the summer in Lake Temescal. Both swim beaches stayed open through the swim season without HABs closures. And at Lake Ansa, this happened the last time in 2018. The phosphorus levels were low until the beginning of the storm season. And then from the from the phytoplankton analysis, we know that both lakes have a production potential. They're both eutrophic. The treatments reduce that potential, but have not eliminated it. The phytoplankton mass has gone up and down during the 6-month study with notable diatom, dinoflagellate, and green algae blooms. But the cyanobacteria have remained a minor component of the total phytoplankton biomass. And it seems like that the treatment therefore stemmed the dominance of the cyanobacteria. We also have to admit that the 2025 summer was unusually cool, so that might have been contributed to the success. And what are we going to do for 2026? We're going to do a similar approach, although we have no guarantee to see the same outcome. One lesson we learned is that aquatic weeds love clear water. That was a disadvantage of the water clarity that we created. So at Lake Ansa, we saw a huge uptick in milfoil and pondweed. And at Lake Temescal, we had enormous filamentous algae growth. So there's a need for additional intervention. One thing I wanted to share with you that was really sweet, but I don't think I'm able to play the song for you. There was a kids group camp over the summer at Lake Temescal and at Lake Ansa, and they were so happy that they could swim again and that the water was so clear that they actually created a song about Lake Ansa. And the lyrics are, "This is the lake that holds us together, Lake Ansa, the lake that will last forever. This is the lake that holds us together, Lake Ansa, the lake that is now clean." And that was really, really awesome to see how happy those kids were. Oops. So I want to finish with some acknowledgements. The monitoring program to examine the effectiveness and longevity of the treatments was carried out in collaboration with Dave Karen from Blue Water Science, Bill Taylor, and Hugh Dalton. I also want to thank Byron Fuhrman with Clean Water Help for all the chemistry advice, and Eli Kirsch from Lake Tech for the installation of the OST system and the application of the products, and for the awesome graphs that I can use to show you this presentation. Thank you.
Amazing presentation. I love the lyrics. If there's a video online, if you could post that the link in the chat or something, I love that. I think we have time for maybe one, maybe two questions. If folks just want to raise their hands. Okay, I see a raised hand. Sarah, go ahead.
That was a great presentation and just amazing to see the results after that treatment. My question is, when you showed that slide that had a lot of aquatic weeds after the treatment, would you consider removing, manually or through, through mechanical harvesting, removing those weeds rather than doing any treatment or allowing them just to die and fall back down and keep the, keeping the nutrients in the system? But if you do manual removal, you're removing the nutrients. That's something we've done on Clear Lake shoreline in Big Valleys area. Just wondering if you would do that for this coming year.
So that's a great point, Sarah. So maybe I need to connect with you on this, because the problem is we have Western pond turtle in one lake and potentially red-legged frog in the other. So we don't have the permits in place to use a harvester at this point in time, but maybe we can connect on this. Ideally, this would be our approach, but because we currently are not allowed to use a harvester or mechanical removal, we will have to go with a herbicide application.
Because of CDFW limitations? Okay.
yeah, that's what our permitting crew tells me within the East Bay Regional Park District. I don't know the details, but yeah, yes.
Yeah, I'd love to follow up with you on that.
Yeah, please.
Thank you.
And Christina, I see you have a raised hand.
Yeah, hey, this is Christina from the San Francisco Water Board. Thanks for sharing this. My kid is one of those campers, so I'm very appreciative that he got to swim this summer. So are you thinking you're going to have to do this annually? And do you think there's any kind of long-term effects by having to do it annually? Like I was wondering if you saw any wildlife impacts. We've got a lot of birds, river otters, rainbow trout, stuff like that.
No wildlife impacts at all. So what we're going to do this year is I'm going to go out next week and collect some sediment samples, and we'll see how the sediment changed, how much phosphorus is still available to be released. I mean, now we got the oxygenation system in it, so we shouldn't have an oxic release anymore, but there could still be some phosphorus that could be released otherwise. So based on that, we will determine the dose for this year. And I am hoping that after this big treatment, we could have some sort of a lower maintenance dose, but that has to be determined. So you can contact me later in the year, or I can give updates as we go. We're hoping that we can just go with smaller doses for the next years to just call it some sort of a maintenance dose.
All right. Well, thank you so much, Anya. We've got to move on to our next presentation. Sure. If you have more questions for her, feel free to drop them in the chat. And
sounds good. Thank you.
That would be great. Thank you. Okay, everybody, we're going to move on to our subcommittee updates now. Starting with illness work group. I'm going to share my screen for you guys. Shannon, are you with us?
I am. Thanks so much. Happy New Year, everyone. Thanks for the opportunity to just give a quick snapshot. I anticipate that we will be giving a more detailed update along with updates to our respective tracking websites and outreach materials later on for you folks in the spring. But here's a quick snapshot. So again, these are the 6 members in the 4 agencies that represent the HAB-related illness work group. And again, just wanted to extend a real note of gratitude for all of our regional and tribal partners for supporting the efforts. We couldn't do it without you. Next slide, please. Quick overview. So these are the cases that ended up being reported as HAB related to CDC's One Health HAB system, known as OHABs. So on the freshwater side, there were a total of 12 human, a couple dogs, and then fish and wildlife cases. I do want to note that some of those fish and wildlife cases, even if they were a singular group event, were quite impactful, as was noted by some of the regional reports. On the marine side, we're still in process in filing those cases. Unfortunately, it was a horrible year to be outdone by 2023's year, where we had over 2,000 individual marine mammal impacts with our pinnipeds that are mostly represented by California sea lions, as well as dolphins and whales. So we're still working on reporting those. We did have a handful of human cases, most of them surfers and water contact from dinoflagellate blooms in Southern California. And then we have, again, as just a particular impact with the domoic acid bloom that was persisting for 5-6 months earlier this year as the main culprit for these marine mammal impacts. There are 2 open cases that we haven't finished determinations on. So those will be reported out to the regional coordinators when we have those results for the case determination. Next slide. As we did last year, sometimes it's helpful to just kind of see the distribution around the state and the different regions of those cases. Again, these are the cases that were reported to OHABs as determined to be HAB related. I did want to call out in particular Region 5, and this was so eloquently described in detail with some of the different components by Sarah Ryan today as the hugely impactful late summer, early fall fish kill that occurred at Clear Lake. So just wanted to note that in Region 5 here, even though there are a similar number of cases to those, for example, in Region 1, some of these cases are reported as group events that could be lake-wide and absolutely catastrophic, such was noted at Clear Lake. Karen, did you want to have any other comments on that?
Yeah, I guess I'll just add, I mean, Sarah did a great job of summing it up. I'll add that in that case, there were obviously multiple things going on, and just wanted to reiterate that sometimes we do report cases as HAB associated if there are, even if there are multiple things going on. So in this case, there was that Seneca cystis bloom happening as well. So that's why it's reported, even though we also suspect there was that lake turnover event that caused hypoxic water as well. So just wanted to clarify that.
Thank you so much. And that's all we have today.
All right.
Thank you.
Oh, did you want to do this slide quickly, Shannon?
Sorry, one more. Just as a, sorry, apologies. This is a big one. So obviously we really are grateful for our partnerships and engaging with all of you in the different regions and the tribes as an extension of our messaging efforts. But I did want to highlight some pretty significant ones from the last years, including a new FAQ brochure. There's the link there. And this is a great thanks to California Tracking for funding this effort, and then Waterboard as well for taking the lead and putting it on their website. So that's a great FAQ for sharing as an outreach material at various places throughout the state on how to report HABs, what do they look like, what are the types of illnesses that can occur, and how to best protect yourself, where to get more information. As always, we have our updated web resources. So currently those cases show through 2024. We'll get those updated probably in the spring for 2025 cases. And those are offered for freshwater and marine in both English and Spanish. We had engagements with several county health departments. Sometimes they were partners for investigating specific cases. And so those are listed here. We had a few state health departments reach out this year, as well as our federal partners and including some foreign governments, particularly for the significant marine bloom event that happened off the coast of South Australia. And as we have found, some of our best opportunities for outreach and engagement are these training webinars and presentations. And this is just a handful here because we fully acknowledge that our partners have an extension of those efforts in all the individual trainings, particularly Carly and Marissa for all of the new benthic protocol trainings that have gone on, and just opportunities to onboard new partners as we encounter these HAB events throughout the state. I cede the floor to you, Karen, or anyone else from the illness work group for some closing thoughts if they have them. That's all I have.
Thank you so much. Okay, on to drones.
Hi everyone, this is Allie Lee from SWRCB, and so I am the co-chair of the Drums Work Group along with Jamie Smith, also at SWRCB. And so we're a group of folks that are interested in exploring aerial monitoring for assessing HABs in inland waters. And so right now our main focus is drones, but we possibly would be interested in expanding into satellites, especially if it relates to drone works. And so when we say inland waters, we do include lakes and rivers, and we're interested in looking at the different types of indicators that aerial monitoring can be able to look at. And so like algal coverage or maybe hyperspectral. And we're also interested in looking at the technical details. So how do you do it? What kind of drones? Maybe even like data analysis software involved. And so we're a fairly new work group. We just started last year. And so for 2025, we did have 3 really good talks on different topics about using drones. So using drones for algal coverage in Lake Tahoe, a very extensive talk about the technical and logistical considerations of how to do drone, what type of drones to get. And finally, we also had a talk talking about using drones not only to monitor, but also sample using the drone. So that was really interesting. Next slide, please. And so moving into 2026, we are transitioning into a hybrid meeting. And so our group members are starting to do some literature review on existing literature about drones monitoring HABs. And so The meetings that we are going to have in this year will include reporting out on the literature review results, but we also hope to get some folks to present seminars on their work, and so it'll be a hybrid of the two. And we haven't really talked about the exact frequency of the meetings, but we expect maybe like around every 2 months or so. And so if you're interested in any of our seminar recordings or even just joining our reporting out, or if you're interested in helping out with the literature review, please contact me or Jamie. And so that's all I have.
All right, thank you so much. I think I am up next. Karen Odkins here again. I am one of the co-leads of the Fish and Wildlife Work Group with Alvina Mahinto as my other co-lead. So our overall goal in the Fish and Wildlife Work Group is to assess and address the impacts of HABs on fish and wildlife. We have some current long-term goals, which is mostly to develop the scientific basis for the aquatic life criteria. And that's something that water boards typically would be putting out and many agencies would use. So in order to kind of get there, we don't really have the data yet that we need, so we're trying to kind of lay that scientific groundwork. Right now, our work group has kind of the structure of a primary work team, and then we have a larger team that's going to review our products once we come up with them. So kind of the flow that we're going to go working through is first we're going to identify some of the data gaps in the literature. We're going to fill those data gaps, and then we're going to propose and advocate for those thresholds. So, we're a pretty new work group too. We had our first meeting this year, but already we have done kind of a literature review starting from Alvina Mahinto's paper that was released in 2021 and kind of looking at what research has come out since then and collecting all that data. And then now we are working on kind of thinning that data to make it specifically relevant to California and what we have going on here. And we're also been, trying to identify some of these data gaps. And we're going to, we've outlined our paper, we're going to be writing up those findings, and we've been meeting with various agency staff to try to figure out exactly what they need in order to make those thresholds. So we're working with them closely from the start. So in 2025, we met for the first time, like I said. We discussed what scope of work we kind of want to do initially, and we narrowed it down to that freshwater aquatic species microsystems, just because that's what data is most available right now. So it's kind of the lowest hanging fruit for these kinds of thresholds. Eventually, we'll probably want to move on to some other toxins and, different exposure routes as well. But that's kind of in the long term there. So like I said, we conducted that literature review of relevant dose response studies, and we outlined our paper. Next steps for the coming year, we're going to be thinning that data, like I said, for being relevant to California specifically. And then we are meeting with water boards to try to understand their process better and what they need in order to create these kinds of criteria. We are writing up that literature review, and hopefully we'll get that published. And then if, things are going well, we're hoping to start applying for grants so that we can do some of those lab studies that we need in order to have data and move things forward. Thank you so much. If you're interested in joining, feel free to email me, and that's my email there. Okay, on to instrument subcommittee.
All right. My camera's on the wrong one. Here we go. Here I am. Okay. Hi, I'm Emily Richardson. I'm a senior environmental scientist at GWR and one of the co-chairs of the CCHAB instrument subcommittee alongside Nick Hampstead from USGS and Cynthia Rice from the water boards. Next slide. So we are a newer group. Sounds like there's a lot of new groups popping up, so that's pretty cool. For those of you who don't know about us, our aim is to connect people who want to learn about all things related to CHAB instrumentation and data collection methods. Next slide. Perhaps our biggest 2025 accomplishment was holding our first ever meeting. We then sent out a member interest poll, which guided the meeting presentations based on that feedback. And we've had many great presentations, including core flow barometer interference testing, brand comparisons, an overview of commercially available nutrient analyzers, even HAB detection with acoustic instrumentation, which was really interesting. And we've had a subcommittee formed from this group called the MDI, or Microcystis Visual Index Technical Advisory Committee. And they are working towards method standardization and improvements for the MDI, which, if you haven't heard about it, it's a visual scoring 1 through 5 of how concentrated a water body is with cyanobacteria, not just microcystis anymore. That's a bit of a misnomer. But this committee is working with a group who's also making an app that will automatically score the water for you, and it'll send results to a live dashboard. So it would be really great to get more people across the state involved in that, not just Delta people. Anybody who has cyanobacteria in their waters, which is everybody here, really. Next slide. So for this coming year, we will embark on that MDI methods testing project. Please email me if you're interested. It's going to be a no-cost participation. We've got supplies for you. So we'll also hold a lead training for this MDI project in the spring at the IEP workshop in Sacramento, but we can certainly set up other training opportunities if there's interest. And for the instrument subcommittee, we have a lineup of more really great presentations, including using solid standards to calibrate the FLUORO-probe, which is essentially the FICO probe, which sounds like a lot of people should use. We'll have information on eDNA auto samplers, presentation on telemetry, field microscopes, and more. So there's my email. Let me know if you want to join. We meet the first Tuesday of every month from 10 to 11, and we are a nice, fun group of people. So, yep, let me know if you want to join, and that's what I've got.
Thanks, Emily. Great opportunities for folks. All right, mitigation. You?
Hi, good morning, everyone. Thank you so much for your time today and the opportunity to talk with a few updates on the mitigation subcommittee. My name is Hugh Dalton. I'm the subcommittee chairperson. It's been leading the show for a couple of years now. Just a little background from the slide you can see. We're essentially a small subcommittee group focused on managing and mitigating HABs in inland waters across the state of California. During our meetings, we share and evaluate emerging mitigation technologies, assess field applications to reduce HAB impacts, and develop guidance documents to inform mitigation decision-making. We also have a round robin of updates from our attendees, be them lake managers, lake consultants, lake applicators, state water board, and other members of our committee. Most recently, in the last few months, there's been a lot of influx of interest to my group. I've got Jamie Smith joining soon in March when we begin. I've got Lori Weber from the Water Quality Council joining in. I've already got Guru Chand from the Aquatic Pesticide Application Group for the state of California that's been with us for about a year, as well as my co-chairs, Dave Karen and Bill Taylor, and to highlight our work with Anya Bray today. What a great presentation, Anya, and thanks for your, giving us the nod for helping with both education of the monitoring plan, helping Anya with limnology understanding, and just really working with other consultants to make Temescal and Anza a better place to swim and visit. Our group meets on the third Thursday of each month from 2:30 to 4 p.m. during the months of March through October. We do take the winter off, not necessarily to stop working, but our work group has a lot less to talk about during the winter months when HABs are a little more under control. I've got a couple more things to mention. I made a short list today for you of a few core sciences that are behind lake management and mitigation success, listing in alphabetical order: aquatic biology, chemistry, ecology, economics and sociology, engineering, environmental science, fisheries science, fluid dynamics, geology and sedimentology, GIS and data science, hydrology and hydrogeology, limnology, meteorology, microbiology, and physics. This is a big list, and I think that it's part of why such an in-depth understanding of at least understanding some of those sciences, and then for the rest of us being intimately close to the science of our interests that we've studied in college or utilized throughout our water careers. It's effective to know a little bit about that and to learn more about that, and just it increases your success to understand what goes on. We talk a lot of technical talk at my meeting. It's always a good discussion, and if anybody here today would like to join, there's my contact info down below, email address.
All right, Hugh, thanks. Did you want to talk about anything for 2026, your plans?
Okay, well, our last update on the website kind of listed some kickoff in 2024. And then, so I think that last update was last year in 2025. So we joined on with East Bay Parks last year in 2025 as they asked us to come in and help evaluate and work on their lake mitigation. In 2024, I've started volunteering with the County of Santa Cruz and the City of Watsonville on Pinto Lake. Pinto Lake is an 8 to 10,000 year old natural lake on the central coast of California. Its degradation was obvious, occurring in the 1960s with the beginnings of toxic algae, and from the 60s, 70s, 80s, 90s, 2000s, and now we're in the 2020s. So for 60 plus years, things have been difficult to mitigate there. Part of the issues that we're revealing in our evaluation are the pollution runoff coming into the lake, and also the modification to the natural lake that occurred in the mid-50s when they increased the dam. They took it from a natural lake to a dam enhanced lake by building a dam and an overflow pipe, and it really increased the sediment accumulation and the pollution accumulation, having changed that once natural lake from easy inflow outflow, no lack of that, to the impound that it is today. And I think that as much as we wanted to really kick off things in 2024, we didn't really get the message across through working with the county and the city until last year. And so this year in 2026 is really going to be year number one, working closely with them, carrying out a plan, getting permits in place, and making those changes. So in the year to come, I will have some good updates with their approval. I can talk about data and give a better presentation, and maybe that'll be as soon as our next meeting in April.
Amazing. Thank you so much. All right. Alan, are you on yet? Stop sharing for a second here. Okay, I know our next presenter is running from a class he was teaching, so maybe we'll just switch. If you don't mind, Carrie, would you mind going now and we'll just switch you?
Oh, sure, that's fine.
Okay, awesome. Thank you so much for your flexibility.
Yeah, are you gonna do the presentation?
No, I think you said it would be easier if you shared, right?
It doesn't matter. Let me pull that up if you prefer.
Or if you want, I can pull up your slides too. I have them.
let's see. There is my, sorry, I'm having trouble finding the sharing.
Okay. I will also introduce
got
you.
it. Let's see.
So we have Carrie Kowalski joining us from San Diego Regional Water Quality Control Board. You guys have already heard from her in this meeting, but she's going to share more about her Lake Henshaw management, HABs management project today. Awesome. We can see your slides, but they are not on presenter. Oh, now they are on presenter view.
Does that work? Okay.
Looks great. Thanks.
Great. Yeah. So Karen had reached out, asking me to talk a little bit about the HABs management, the activities that are going on at Lake Henshaw. Since Vista Irrigation District so kindly volunteered to let us use them as our trial run for the FICO probe that was purchased by State Board. And also because it's an interesting story because starting in 2020, Lake Henshaw was kind of our joke as our like problem child that always had blooms that were toxic. But since then, it has significantly improved. So talk a little bit more about that. So Lake Henshaw, it's located in Santa Isabel, California, which is in Northeast San Diego County. So essentially what that means is it gets really hot in the summer, and it does get cold in the winter because it is at a higher elevation, and they actually do have snow. So they're dealing with quite a range of temperature, fluctuations. So Vista Irrigation District, they own the dam, the lake, and many acres of land surrounding it. And they pump groundwater to augment surface runoff in the drier years. And the local water, of course, is important for many entities. It's used by Vista Irrigation District, the city of Escondido, and then multiple tribes. The lake water is released downstream into the San Luis River, and that provides in-stream uses as well as groundwater recharge for tribes. But then ultimately it flows to Lake Wolford, and then it is treated for drinking water at the Escondido Vista water treatment plant. But again, I guess I want to emphasize the fluctuations and of course the amount of rain and then the temperature. So the storage at Lake Henshaw is quite variable. So at the bottom, I just pulled two numbers. Early in 2025, the storage was over 21,000 acre feet. And then this year, unfortunately, same date, January 1st, only 5,000. So it makes their treatment and management quite challenging. But Vista Irrigation District, they do a ton of monitoring, and they share a lot of information with us, and we work together on some of it, but they do the bulk of the work. They monitor within the lake, and they do have monitoring within well fields and the groundwater and the released water. But for this presentation, I only wanted to focus on actual in-lake monitoring. So on the right, you'll see a map that shows just a few of the stations, and they're kind of the main core stations that will be highlighted a little bit later in the presentation too. So in the white, that's at the buoy line closest to the dam, and then two other sites that are pretty regularly monitored are a little bit further south in red and green. And so there's FD, which is a fishing dock, and then S is shoreline, where a lot of the recreators are. And so it's a big variety of parameters that they monitor. So dissolved oxygen, temperature, nutrients, kind of the typical. Chlorophyll A, cyanotoxin concentrations, they do microscopy, so they get species ID and cell counts. This year we tried out the FICO probe to add to that information. And they really do use the SFEI HAB satellite a lot. It tracks really well with what they actually see on the ground, which is pretty cool. And it shows up, extremely well unless it's overcast. And then they also do have information from an algae tracker, which is a solar-powered sensor buoy, and that takes measurements such as the water temperature, air temperature, turbidity, chlorophyll, and phycocyanin. So they get quite a lot of information. So I'll talk about their adaptive management. So they began treatments which were mostly peroxide in the spring of 2022. And then in 2023, they shifted more to a regular treatment schedule. So biweekly peroxide is mainly what they do, only during the HAB season at Lake Henshaw, which typically is from April to August. They also use a lanthanum modified bentonite clay treatment. That's typically once in the springtime to try to bind that phosphorus into the sediment. And then as sparingly as possible, they will use a copper treatment. However, they really haven't had to use it recently, and the last time that they did a copper treatment was in June of 2024. And they're doing pretty good with all of this, but they still are working on, additional measures. And right now they have an oxygenation system pilot study that was supposed to be online sooner than it could, but it's under development right now. And the Vista Irrigation staff wanted me to emphasize that, this is a learning process for them. They're still learning. It's not perfect, but they're doing pretty good. So here's just a snapshot of the treatments from 2025 from the spring through September.
Are accurate. And so
And it's mostly, so it's the biweekly peroxide, and then you can see the lanthanum treatment in early spring. And I just kind of threw that up because you can see it fluctuates quite a bit, depending on the water levels and temperatures and things. So there's a lot going on in this graph, but I thought it was pretty neat to show. So I'll walk through what it is. So across the top, you'll see the green, which is designating the shoreline station. Orange is the fishing dock, and then the gray is the buoy line station. However, the trends are pretty similar through all those sampling stations. And on the y-axis, you'll see the microcystin concentration. So that was the main toxin that they were having trouble with starting in 2020. And it is shown on a logarithmic scale. But what you'll see is the red line for the danger, CCHAB trigger level, the orange for the warning, yellow for caution, and then the dashed line shows the method detection limit. So the main takeaway from this is starting in 2020, back when we were joking about it being a problem child, we were hitting danger level with microcystins quite a bit. And then you see the reprieve during the cooler months, and then the spring, pops back up. So 2022, you see just the 2 peroxide treatments and 1 copper that they, when they were starting this adaptive management. Then more into 2023 and then beyond is when they do the biweekly peroxide treatment. The lanthanum sometimes, copper every once in a while. But the good news was since end of 2023, early 2024, they've been able to keep all of the microcystin toxin concentrations below. Actually, they mostly have non-detects most of the time, which is quite amazing. So doing a great job with that. And then very similar, same plot for the antitoxin A toxin concentrations. So I don't really need to go into too many details about that. But again, it was showing up in early 2020s and then pretty much non-detect ever since. And then here's the part that we added on our boat outings with Vista Irrigation District. And so here's the information that we get from the FICO probe. So you'll see green algae concentrations on the green line. The cyanobacteria concentrations are the blue line, which is all the way toward the bottom. And diatoms are shown in brown. Those are all from the FICO probe, and then grab samples for chlorophyll A and the phenophytin A concentrations. So I guess hindsight is a little bit 2020. It would have been great to have this instrument. I think it would have been much more interesting if we got to see it during when Lake Henshaw was a problem child versus now when everything's a little bit dampened. But still, it's nice to see the concentrations and how it shows that pretty much now they have more issues with the green algae versus the cyanobacteria. And the patterns that we see, I don't think that we don't really see a huge I guess, effect from the biweekly treatments, because we only took our samples once per month from May through October. But you do kind of see the pattern follow maybe the temperature regime. So that was the buoy line, and you see something very similar at the fishing dock. So the green algae is more dominant than the cyanobacteria there at the bottom. And then the shoreline shows almost the same thing. It's shallower, and we could definitely see a pretty large like diatom community, which is prime habitat for diatoms. So those pop to the top, especially during the warmer months. And then I thought this was kind of fun to look at. So over the years, we did have the species ID and concentration. So you could kind of see that the trends that happen with the treatments over time. So initially, Planktothrix, that was in a high abundance early on from 2020 through 2022, and then disappeared. Dolichospermum was off and on. It's gone now. Aphanizomenon, same, came and went, but then it really wasn't around after 2023. Microcystis was highest in abundance at first, but went off the scene in about 2023. But now, so the species toward the bottom are what we're really seeing. So Snowella seems to be one of the major players. So we had that in 2022 and 2023, disappeared for a long time, but now it's back in 2025. Phanacapsa, that one hung around most consistently, and we still have it there at Lake Henshaw, and a few other species came on the scene in 2025. And I think with that, that's all I have.
Great. Thank you so much, Carrie. I think we're running just a little bit behind. So if folks have questions for Carrie, if you wouldn't mind dropping them in the chat, and Carrie, if you could answer them there, that would be great.
Sure.
Okay, thanks. Okay, and next we have Alan Wilson, who's joining us all the way from Alabama. He works at Auburn University, and he's going to be telling us about using meta-analysis for water resources management.
Great.
So yeah, Alan, if you can share your screen.
Yep. And hopefully you'll be able to see what I want you to see. I know my office is a disaster. I apologize. But it's what it is. And I just actually got out of teaching meta-analysis. So anyway.
Yes, we can see it. Looks good.
Okay. All right, good. So I'm going to go, and I think I know how much time I have. If I talk too long, you can just turn me off. It's fine. But thanks for this opportunity. I'm thrilled to be here. And I kind of wish maybe I was able to go see y'all in person, but this is fine. So just I like to always acknowledge folks at the front end, just because by the end of this, you won't be listening to me. So I need to acknowledge the people in my lab, my students, and people who've been in the lab, people who are currently in the lab. I got a great group, and they work hard. We also work closely with collaborators throughout Alabama, but also throughout the Southeast. We've been working a lot with catfish farmers, so you're going to see some beautiful pictures of green ponds, which should kind of amaze you. They amaze me when I go see them. But we also kind of work with drinking water utilities, which don't have nearly as bad a water. We work closely with USDA folks, and of course, Auburn lets me do lots of things. So I got to acknowledge them. And then funding, all this is possible because of money we've gotten from NSF and USDA. So this is just kind of how I see my life, the work that I do. So I'm an aquatic ecologist. I've been studying algal blooms for a long time. I do meta-analysis, and I just recently, and I do a lot of work with undergrad researchers and also high school researchers. And I was actually just able to publish a study that kind of blended all these pieces together. So kind of in the center here, if I can, I don't know where my little pointer is, but let's do that. So this space here, I actually have some coverage, which is exciting. And at the very end or near the end, I'll talk about, I think, what y'all really wanted me to talk about, but I promise I'll get there. So just bear with me. So I take a lot of pictures. I love looking at green ponds. And so a lot of people are freaked out by these things, but I love them. So just to show you, I study aquaculture ponds that often they all have aerators like this. They use at night. I do a lot of like remote sensing work with drones, and so this is just one picture of a farm that I visit often. This is a catfish farm. They happen to be harvesting fish down here on that particular day. We then take data from these drones, and then we do things like calculate these vegetation indices. And just to give you a sense of how different these ponds can be, so there's 19 ponds that we sample here. You can see the little dots where I actually go sample, and then now, I'll fly my drone and I'd run around and get samples and then try to see how the data match up. But so this was in February, now about 2 years ago. I go out once a month, and to give you a sense of what this might look like later in the year when there's a heavy bloom. But you can see like some of these ponds, there's not, and there's really interesting things happening in some of these ponds, which we're trying to explore. And this one particular pond over here, which has got like real tons of heterogeneity. This is one that we've been spending a lot of time with, trying to help the farmer produce a lot of fish and not have issues with algal blooms. Again, I like to show pictures. So this is our field station where we do a lot of research. We do a lot of field experiments. That's kind of a thing that I love to do. If the students want to get me excited about anything, they just have to propose a field experiment. So that's what's here. This is another one. There's my students that are sampling. This particular experiment, we were doing different types of chemical kind of algicides, and you can kind of see some of them working differently. And so those colors are indicative of the treatments. There's of our field stations. You could just, again, I love being outside sampling. So the reason why we do this, at least for the fish farmers, is for situations like this. This is terrible. Those fish are dollar bills that are dying or have died, and the farmers don't like this at all for a lot of reasons. But so my job is to try to help them figure out what's causing these fish kills, and if water quality related to algae is part of it, to see if we can solve that problem. So this is what I'm going to talk about today. I talk kind of fast, kind of mumble. I apologize. At the end of this, I'll have my contact information. So if you need to reach out to me, you're welcome to. Also, on where I talk about papers, there's like a QR code. So if you're interested, you can just click that and it'll take you to the paper. So you get more information. But the goal today was to talk about meta-analysis, and there's a few examples I'll talk about. The first two I'll talk about briefly, and the third one is, I think, why I'm actually talking to y'all today. So I'll talk about that a little bit longer. And then actually I'm going to feed into the very last thing will be an experiment that was based on the results from that meta-analysis. Okay, so the first thing I'll talk about is actually a meta-analysis I did as a PhD student. And this has been a long time ago. I'm old, but at that time, I think the paradigm was that cyanobacteria were toxic to zooplankton. And we just weren't really seeing that in the data we were collecting. And so we decided to do a meta-analysis to just see, based on lab studies, feeding zooplankton, primarily cladocerans or rotifers, diets with cyanobacteria, how did the zooplankton respond? And we were using population growth. And again, this is the title of the paper. And again, we were looking at toxicity and morphology. Again, I'm not going to go into the details about cyanobacteria. You all know this, but we thought that morphology might matter because little algae might be easy to eat for some zooplankton than big algae. And then for toxicity, of course, I've heard, the earlier talk was talking about microcystin. We know these things could be potentially harmful. And so the idea doesn't matter. And so we were looking at experiments with known toxin producer cyanobacteria. Really simple design. We looked at population growth. That's what this little R is. The RT is for the treatment. So it would be a diet containing cyanobacteria, any percent from 1% to 100%. And then our reference was a good diet of some good alga. So probably like usually it's green algae or some cryptophyte or something. And so it was just a simple comparison of the cyanobacteria containing diet versus a good diet. And the way that this works, here's the formula, is that if you get higher numbers, that means the cyanobacteria are good, or at least better food. And then when the number is lower, it means the cyanobacteria are worse. And then when you have a number that's above 0, it means actually the cyanobacteria were better for that diet was better for the zooplankton than the control diet, which is kind of unusual. We don't see a lot of that, but that is a thing. So the first figure is looking at the amount of cyanobacteria based on carbon in that particular diet. And as you might expect, as you increase cyanobacteria, the effect gets worse for the zooplankton. So that's not a surprise. But you see this range, and I think this is what's interesting, is there's a large range. So some zooplankton don't seem to care, some care a lot. So there's a lot of variation there. When you look at the size of the zooplanktons, it actually seems like it doesn't really matter at all. And at least early on, we thought that maybe big zooplankton could avoid some types of cyanobacteria because they're maybe too big and they're not seeing small cyanobacteria. But then there's a lot of big cyanobacteria. So the fact that there wasn't much of a relationship with grazer size was interesting to us. When you look at all these data together, what you find, which I thought was actually interesting, is that toxicity doesn't really matter, at least based on these data. And if you look at all the data we have, this data point right here shows that cyanobacteria in the diet are bad. When you look at the effect of toxicity, there's no significant effect. And if you look at morphology, it does seem like there's a difference. And maybe surprising is that filamentous cyanobacteria are better, or maybe not as worse, or not as bad as single cell and colonial. And what we kind of thought was that if you have a big filament, maybe it's hard to eat. So that maybe is one defensive mechanism if the cyanobacteria are too big, then the zooplankton don't eat them. Again, the data down here based on cladocerans or rotifers. And so you can just see, like this finding here to me was surprising that toxicity did not matter. And the more I do these studies, I think this is probably true. I don't think toxicity matters a lot, at least in the studies that we've been running. We took that same data set and we were interested in looking at like species level effects. And so this figure here is looking at different types of zooplanktons and how they perform on non-toxic cyanobacteria or toxic and on these effect sizes. And you can see typically non-toxic are not as bad as toxic generally, but in some cases you see toxic cyanobacteria actually seem to be maybe a little bit better, not statistically significant, but maybe a little bit better. And it's hard to see, I know, but if you look at this particular figure here, this is actual population growth rate data. The thing that I wanted to really point out here, which I didn't actually pay attention to this when we wrote this paper, but later it became clear that if you look at those data, a population growth rate above 0 means that the zooplankton are growing. They're growing slowly, but they're growing. And what you see is that in almost all cases, the zooplankton grew positively on cyanobacteria, some percent of cyanobacteria in the diet. And again, I think if you think about that, that suggests that maybe zooplankton can eat these things. They just need time. So anyway, those two papers kind of came out of that one meta-analysis. And I don't know if the paradigm is the same that when we see an algal bloom, it's necessarily because the zooplankton are being killed because of the cyanobacteria. So the next meta-analysis is actually one from a student who graduated from my lab a few years ago, and he was interested in kind of synthesizing all the data out there that was available. He created correlational data, and maybe I think the next figure shows this. So this is the title of the paper, and he was basically trying to get correlational data of microcystin and parameters that are available, like nutrients or algal abundance or whatever. And there are the data sites. So you can see a lot in North America, but some in Europe and Asia. And so these are the data. These are correlation coefficients. So again, the range goes from -1 to +1. The things that I wanted to point out, they're physical parameters here. When you see that these 95% confidence intervals are away from this null, it means they're statistically different. And I guess that's what these little asterisks mean as well. The important part about these figures I'm going to show you is that there's not a lot that's statistically different. And that doesn't surprise me. It's kind of frustrating, but it doesn't surprise me because we know toxicity is kind of messy. But temperature, again, warmer systems have more toxicity. That's typically when cyanobacteria out there, so that doesn't surprise me. Again, Secchi depth is, more algae, you get shorter Secchi depths. And so these things matter, but turbidity, TSS, conductivity, no clear patterns. And then when you look over here, there's just not a lot of statistically significant effects. And I think that's really interesting. Of course, algal abundance is important, but most of these parameters are not statistically significant. And I think the nutrients are here. Again, there's a bunch of different parameters. Almost none of them are statistically significant. And, the ones that are might surprise you about phosphorus being positively related to microcystin. Microcystin contains nitrogen, so maybe you'd think that nitrogen would be important. Nitrate down here seems to be significant, beryllium, as well as dissolved nitrogen. So the point of this is that despite all the data out there, it's still pretty messy. And I'm not sure if this study helped except to maybe reinforce that. Okay, so this is the study I think I was asked to talk about, and I'm going to talk about it. I'm going to go into more detail, so if you don't have a lot of familiarity with meta-analysis, you're going to get more of it right now. Sorry, I blasted through those others, but this is going to kind of talk through a little bit about what we did. I wanted to highlight Saranya. So this was the lead author. She was one of these superstar high school students who reached out to me during COVID and wanted to work with us. And I asked what she wanted to do. And she had these ideas that were like, would cost like $1 billion and need like a vessel for like the ocean. And I was like, we don't have any of that. So I was like, let's do a meta-analysis. I've been wanting to do this one for a while. So she took a graduate level course, the one I'm teaching now. She took this course remotely. And she came to work with us. You can see her sampling, but she was amazing. So I just want to do a shout out to her. Working with high school students is not easy. Working with any students is not easy. Working with high school students is tough, but she worked hard, and I value that. So so the databases we searched were these three. We were looking at keywords. We're trying to find studies focused on field experiments that use some kind of treatment to kill algae. That was kind of what we were looking for. That's what you can see down here. We found 39 papers that were included. We found a lot more, but that's what we used. The way that we dealt with these data, so if you're interested in meta-analysis, is kind of what we did. This is an example of what the data would look like. So then you're asking, how do you get data from these kind of studies? You actually have to digitize it and go get the data. So we get mean responses, error around those means. The sample size is buried down there. When the data were collected, and then treatments, of course. And so, and then in this case, there's as y'all all know, I mean, we have a FICO probe like y'all do. We measure chlorophyll like y'all do. So there's like a lot of things that we're doing, but not everybody does the same thing. And so we tried to group data accordingly. And so those are the kinds of things we would get from figures. And then I talked about these effect sizes. In this case, this effect was different than that first study I talked about. In this one, it was just a simple ratio of whatever the parameter was. Let's say it's chlorophyll in the treatment divided by chlorophyll in the control. And we did the natural log of that. So it's called the natural log response ratio. And then the beauty of this is that if you get an effect that these little 95% confidence intervals don't cross zero, it means it's statistically different. Then they're different. If you get an effect that crosses that null, it means they are not statistically different, meaning the treatment was not effective. And then in some cases, we actually saw one where you get a positive effect. Again, that's the wrong direction. So that means that whatever you did made the situation worse. So a really simple way to interpret the results. So one thing that we struggle with initially was like, what are we looking at? Because we have all these data. Could we just use all the data? We have all the data in a spreadsheet, but we're like, we should decide what data we want to use and be thoughtful about it. So what we decided, and I think this was a good idea, and it's biased. I totally think it's biased, but it was a good idea in that we decided to look at the worst case scenario for the response in a sense of where maybe it's the best case for water management. So where the treatment effect is the biggest, and that's what it says, most effective day. And you can see where these treatments are. Those were the most effective days for this particular study, and they'd be compared to the controls. And then we also decided to do the end of the experiment. I wasn't as excited about that because we typically run experiments kind of past their lifespan just to make sure we got all the data, we're good. So I wasn't really confident that would mean much, but we did it anyway because we had the data. All right, so this is what the figures you're about to see will look like. This is called an orchard plot. It's kind of unusual. It's coming to be a thing in meta-analysis, but a lot of people have never seen this. So I'll just orient you. Each little dot here is an effect from a study. The mean is here. The 95% confidence intervals, these kind of thick whiskers. The 95% prediction intervals are the skinny whiskers. Those particular prediction intervals would tell you if there was a new measurement where you would predict it would be. Again, mindful that the 95% confidence intervals are telling you if this thing is statistically different or not. And so this null is here. The asterisk is telling you it's statistically significant. This is the number of effect sizes. This one is the number of studies or papers we got those effect sizes from. And also the size of these little bubbles gives you some confidence in kind of the precision of the results. Okay, so I'll show you the results. Again, we had the most effective day, which was biased, but looking at like when these treatments would be the most effective, what would we see? And then the end of the experiment. So on the most effective day, the treatments showed 70%, that they were lower by 70% than the control. So clear effectiveness, that was awesome. So it does show that these treatments are working when you look at all the treatments. And maybe I forgot to mention, we were looking at biological or like bacterial, plant, chemical, and physical. At the end of the experiment, there was a statistical effect, but it was small. It was only 29% lower for the treatment. So the treatments were working, but they were 29% lower than the control. So not huge effects. But overall, it seemed like these treatments worked, which is good. The thing that was maybe surprising was that when you split these treatments up, again, bacterial, chemical, physical, and plant, the only one that showed significance was the chemical. And part of this is due to the fact that there's a lot more data here. And if I show you the end of the experiment, you can see sample size. So like tons of data were in the chemical. We just didn't have a lot of data for the others. So 88% of the effect sizes were buried in chemical. So you might say that the lack of significant effects was due to just lack of like large sample sizes, and that could be true. But it's kind of surprising. So people are, and I heard about using kind of these clays and things, and I'm not saying you shouldn't use that, but these data don't support that those things are that effective. So then we actually said, well, let's just split all this up and look at each particular treatment by itself. And so again, you can see how they're grouped: bacterial, chemical, physical, and plant. I'll tell you, as you keep kind of narrowing down, the sample sizes get smaller and smaller, and that's just an artifact of what we're doing. The thing that's really interesting when you look at these data is that only 4 of all these things that were tested showed statistical significance with this meta-analysis. And I think maybe only 3 of those showed it on the most effective, or at the end of the experiment. So most effective day 4, end of the experiment 3. One of those, this diquat, actually had a positive effect, which again is the wrong direction, so that's not good. And that was at the end of the experiment. So that to me, this kind of suggests that a lot of the things people are trying are not working well, or we just don't have enough data. So based on these data, so Saranya, she worked with us for several summers. She's at the University of Pennsylvania now, so she worked hard and is in a good school now. And so we talked, I was like, well, let's use these data and come up with an experiment. So I'll show you some results from this experiment. So we decided to look at copper sulfate, which y'all all know kills algae, kills lots of things. And then phoslock, which is maybe that thing that y'all were talking about earlier about this clay that's ideally would bind up the phosphorus and keep it from feeding the algae. So that's what this experiment was that I'll talk about. This paper just came out. And I think it's interesting. So I'll show you the results. This is where we did the experiment. There's our little floating dock. It's a beautiful place to do work. I like to show these pictures to encourage people to get outside and go do experiments. Again, another picture of us setting these experiments up. So this experiment was 56 days, 1,700 liters on that floating dock. We had 4 replicates of 4 treatments, nothing, copper sulfate, phoslock, that clay thing, and then the combination of the 2. I'll tell you that the combination of the 2 used half of what each of the whole treatments used. And so that's what you'll see here. And that actually became really important. A little bit through the experiment, we noticed some really interesting results, and it wasn't something we thought about at the front end, but it looked like the copper was doing some really weird things. And I was like, let's go treat every treatment, half of the replicates with copper again, and let's see if we can duplicate what was happening. And I'll show the data to you in a second. So we added another dose of copper on day 13, and that's what this is for. And then you can see when we sampled, I bold these data because that's when we really sampled. Saranya went back to school, and I don't know, either I was lazy or busy, but I went out and used our FICO probe out in the experiment. So I got some data that I'll show you, but we didn't get a lot of data these later dates, but there was some really cool stuff happening, and I didn't want to stop measuring. All right, so these are the chlorophyll data, so our extracted data showing all the algae, again, based in chlorophyll. This is when we treated with copper. This was that later treatment that I talked about. Again, control in black, copper's in red, phoslock in green, and then the combined is in yellow. And then you can see the doses over here. I work with catfish farmers that dose in ponds based on pounds per acre foot, so you can see these numbers. And the thing that's cool, there's like so many interesting things about these data. The first thing is that when you add copper, it kills stuff. Again, not a surprise. The thing that was shocking, but we see this repeatedly, is that as soon as you treat, you kill stuff, but then they just fire right back. And so you get all this algae, which is really interesting. The thing in this particular case, which was cool, was that where we added less copper, so the half dose of copper, we get the algae goes right back down. Whereas when we added a full dose of copper, the algae just skyrockets by the second week, which is kind of surprising for me, I think. These little blue dots are, sorry, I forgot to mention that, fertilization. The control didn't really do anything interesting. And then the phoslock didn't really do a lot interesting. It kind of matched the control, which maybe some people would think that's not good, but actually I think it reinforces maybe that Foslock maybe was better than copper, at least in this case, unless you're trying to grow algae. So, but the point of this figure is showing that early on copper does what you think, but then not too long after things get not good and you have more algae than you want. When you look at phycocyanin, so we do extractions for phycocyanin like we would for chlorophyll, but a different method. And what you see here is you get these huge effects, rapid loss when you add copper. The thing that's interesting in this particular case is the cyanobacteria disappeared pretty much in all the treatments, at least up to 2 weeks. So if the goal was just to kill cyanobacteria, everything we did, even doing nothing, seemed to be good, which was cool. But we let the experiment run longer. And this is the part I wanted to show, which I thought was really interesting. And this is what I'm seeing now is that we're overdosing copper and we're killing algae, but we're also killing the zooplankton. And I really think zooplankton is what we need to focus on more than the algae itself, because if we can manage zooplankton, we can take care of the phytoplankton. And so when you add, so we added the second dose of copper, and you'll see the triangles. That's that second dose. So these red ones were then split. So 4 replicates now are 2 replicates. That's why it's messier here. But you get, this is a double dose of copper. This is now a single dose of copper. Actually, this is the first dose of copper. So I'm trying to look at this again. So 1 dose of copper, and then this one is 2 doses of copper. So the triangles would be indicative of where we added copper on that day 13. And if you notice that where we see the highest amount of phytoplankton is chlorophyll, it's where we added copper. And this is just generally what happens. Where you're adding copper, you get more algae. Again, the controls kind of hung out where they were the whole experiment. The Foslock did too, but the Foslock kind of over time actually became the lowest, at least for chlorophyll. When you look at phycocyanin that we extracted, again, this is really interesting. So where we added copper twice, we got the highest concentrations of phycocyanin after almost like 4 weeks, right? That's shocking to me, right? That's not what we want to do. So those data I think are really interesting. And then like I said, Saranya went back home. So then I just went out there and measured with my phyco probe, and you can see the results here. So where we extended it. And like I said, I kind of run experiments until they're dead. I stopped fertilizing after she left. And so things kind of slowed down. But the data that I really wanted to show you was over here. And this is why I kept going out with a phyco probe, was because I noticed that where we treated with copper once or twice, we had the worst water. So the cyanobacteria was the most abundant. And again, this is the wrong direction. Like we're thinking that we're adding copper and it should make the water quality better. It is doing the opposite. So that's why I wanted to show those data. Then also, yeah, so this is where I make my speech about zooplankton. And so these are the phytoplankton data, the chlorophyll that we measured. These are the zooplankton data. Again, I didn't go out and get samples later in the experiment when Saranya left, and so we didn't have zooplankton data for the later part. But by that point, I wasn't fertilizing, and the zooplankton were kind of crashing. But you can see, like where we added copper, the zooplankton, where we add more copper, the zooplankton crash. Where we had less copper, they don't do as bad. If we can just find the right dose of copper where we kill what we want to kill, but we don't kill the stuff we don't want to kill, I think that's what we're trying to do. And so where we added a half dose of copper, the zooplankton did fine. I'm a Daphnia guy, so cladocerans are kind of what I think a lot about. And so big, large zooplankton are what I'm trying to promote. And again, where you add copper, these things are just so sensitive. You add like a little copper, they smell the copper and they're dead. And so we just don't see any copper or any cladocerans where we're adding copper, which is terrible because these things are really important for controlling phytoplankton. Then of course, and you'll all know this, but if the algae aren't there, then there's nutrients that are going to get more abundant. So SRP tends to increase where you don't have a lot of algae. And so you see the copper data here. That's because there's a lot of algae, so there's not a lot of SRP. And then I put ammonia in here because ammonia. So this is ammonia and ammonium, but ammonia can be toxic, and so we worry about this. We regularly see where we add copper, ammonia spikes, and again, those ammonia spikes can be lethal. And so I kind of point this out that be careful where you're adding copper because you get these ammonia spikes, and if you're not, if you're unlucky, you could have a big fish kill. So I don't know how I'm doing on time. I'm just talking, but so meta-analysis is super valuable. I don't think enough people are doing this in water quality research. I think there's tons of data. I think there's a lot more room for this. It's hard, it takes a lot of time, but it's something that is done and could be done, and more of it could be done. It doesn't cost a lot. It just takes time. I think that these meta-analysis can be used to kind of show that maybe paradigms aren't really what they are. I mean, maybe we need to rethink about what we understand about these systems. I mentioned copper overdosing. I think pretty much everyone's using too much copper, and I'll encourage people to not use copper, use much less. And again, that's what this last bullet says. So the last thing, I just want to make a pitch. I am teaching this class to people at multiple schools right now. I don't charge money, and if it's just if they want to join me. I keep the class small, but I want to say I'm teaching now. I don't want anyone to join now. But if you or your colleagues are interested in joining us in the future, just email me. And I'd be happy to just keep you on our email list. And we've got people from 4 other schools outside of Auburn with us this semester. And then the last thing, I know there's a lot of people on this call that might know someone who's looking for a postdoc or a technician position or grad school opportunities, but we're always kind of looking for people. We're really blessed to have support to bring in new people and teach them how to study algal blooms. And so if you know people, let me know. But I think I'm going to stop talking, and if there's questions for me, I'll do my best to answer them.
Awesome. Thank you so much, Alan. That's super interesting. I'm sure people have questions, so feel free to raise your hands. Okay. No questions.
Sorry.
Oh,
I
yeah.
have one.
There you go.
Hi, Alan. That was a really interesting talk, and I really loved it. So thank you for giving it to us. I might have missed it, but when you were talking about the last experiment, did you guys look at what the cyano community was in the different corrals? Or
We did. Something that if you ever visit me, I can show you a closet that I have that has like 10,000 phytoplankton samples. So I'm like notorious for getting phytoplankton samples, but as anyone who counts phytoplankton knows, it's easy to put them in a bottle, but not to count them. So we did look at what phytoplankton were in there. I don't remember what it was, but that paper exists, and actually, I'll put a link to the paper in the chat so you can go get it. But I don't remember exactly. But we usually count to see what was in there at the start, and then if we can get away with that, we are fine. Like we trust our extracted phycocyanin data. The FICO probe I think works okay. It maxes out at a pretty low number for our aquaculture system. So it's more of just kind of an indicator how much algae is out there. We also have a Turner Designs cyanofluor too, so we use that some, but the FICO probe is kind of our thing that we measure every sample with, but then we always get samples to run extracted phycocyanin and chlorophyll. But again, I don't remember what algae was there, but I'll put a link to the paper in the chat before I bail.
got it. Thank you. And so, yeah, with all your experiments or experience or like research onto the different treatments methods, or especially the chemical ones, I was wondering if you have any comments or idea if certain chemicals like copper versus like peroxide, like biases what like effects on certain groups of cyanos, or like does it lead to like eventually a bias of a certain community of cyanos?
I mean, so ideally, I appreciate that question, and I'm going to put the chat paper link in there. So hopefully this works. So you can go get the paper if you're interested. So that's a good question, and again, I'm probably going to offend someone, so I apologize. But the point of that question, I think, was if you're going to promote cyanobacteria, a certain kind of cyanobacteria, and ideally we're treating to get rid of the cyanobacteria, I think. And so the fact that we're even thinking that treating could lead to more cyanobacteria makes me question, should we be doing this anyway? So again, I'm an ecologist, so like I think about zooplankton and phytoplankton. So like the work that we're doing out in these fish farms, I'm actually trying to get them not to use any chemicals at all. And I say this honestly, like it's not an easy thing to talk to them about because they like to use their chemicals, and they do work. But what we find is that when you treat these ponds, you kill the algae, but you kill all kinds of other stuff, and these communities just get like reset all the time. And so I think if you could, at least in these catfish ponds, and catfish don't see well, that we've seen this, we've got data that if you can keep little fish, like small, like planktivorous fish like bluegill out of these ponds, the Daphnia get really abundant. I mean, I have like vials, which in my messy office, but like tons of macroinvertebrates, like amphipods in these aquaculture ponds that are so full of fish, but they're not fish that eat amphipods. And so, and the water is not really green. So like I'm really pushing these farmers to think about not using any chemicals, honestly. So that's kind of my bias is I'm trying to get people not to use chemicals at all. We tried peroxide, and it's funny, I told some farmers about it. This has been several years ago. After a week-long experiment we published, and they were all excited. They bought drums of peroxide and started using it. And then we did a longer experiment, showed that the peroxide was terrible. So I don't tell anyone to use peroxide personally. Because you just have to, I mean, all these things are what's happening is you treat, and then the algae go away, and then they come rearing back, and you're treating again every other week or so. And it's like, is this what we want to do? So to answer your question about are certain treatments favoring certain kinds of algae, I think it's probably too context dependent. Like I don't know. I haven't seen that adding copper causes microcystis to bloom because like a lot of our catfish ponds, microcystis is like the dominant critter. So it's like it loves those ponds. Drinking water systems like recreational reservoirs, we'll see a lot of filamentous cyanobacteria, but I don't have I can't tell you like in my synthesis of my simple brain here that one treatment would push a system. I would say the only thing I could tell you with confidence is that if you treat with chemicals, especially if you overtreat, you're going to lead to more cyanobacteria. So I think but I don't know what kind of cyanobacteria, maybe what you had before.
Thank you so much. I do want to be respectful of everyone's time. I see there's a couple more questions. So, Alan, if you don't mind staying on and answering those questions,
Sure.
that
Yes.
would be awesome. But if folks need to go, totally understandable. So I think Sarah was next with her question.
Yeah, let's have it, Sarah.
Hi, Ellen. Fantastic presentation. We need you in California.
Thank you.
Did you, I know we don't have time, but if you could just point me in the right direction, or answer this particular thing of, do you look at toxin levels in fish tissue at all? I mean, is this why you're doing these studies? And where can you show me any information about that regarding your studies?
Yeah, so that's a good question. So my postdoc at Michigan, I worked on Lake Erie. We looked at yellow perch like livers for microcystin, and so that's something that we've done. We, for a variety of reasons, we don't look at fish tissues for microcystin now. And any of you who work with vertebrates know it's kind of a pain to get approvals. So we don't. We've done this in the past. We see microcystin in these ponds, the catfish ponds, all year. Like, and I can show you beautiful data. I've been sampling ponds for like 7 years now, every month. Beautiful seasonal data, clear patterns of cyanobacteria. Microcystin's always out there, but it's really low. Like right now it'll be low, but like by May it'll be booming. Um, I suspect it's probably in the fish, but I have not measured. We've not gotten good data on microcystin levels in the fish in the systems that we study. So, and I'm sorry, I don't have anything to tell you about that.
All right. We have a question from Zach. Do you have any data on whether copper or other chemical algal treatments are harmful to aquatic insect communities?
That's a good question. Like I said, if I walked over, I could grab this beautiful vial that has all these macroinvertebrates. I'm not an ecotoxicologist, but I'm sure there's tons of people who've done little experiments, putting insects in vials and added copper in to see what happens, or bugs. But I don't know. We're trying to publish a paper that might get people to think really more carefully about copper dosing in lakes. Again, we don't. I I'll be honest, like seeing macroinvertebrates in aquaculture ponds kind of shocked me. And because you don't see them, like you have all these planktivorous fish, there's no chance for zooplankton or even larger macroinvertebrates living. And so for me to like take a bucket and kind of push it through the edge of the pond and get like a billion amphipods just blew me away. So I don't know. I suspect, I mean, copper is really good at killing stuff. So I think the doses people typically use, and this is like for recreational users too, I think they're way overdosing and they're probably destroying macroinvertebrate communities, but it's not something that I've studied. And I do know that in these ponds that we work in, if they don't add copper and you can keep the planktivorous, they call them scaly fish away, in these catfish ponds, you'll get tons of macroinvertebrates. So they need a place to live, without fish or copper.
All right. Well, I think those are all the questions I'm seeing for now. Feel free to reach out to Alan if you have more that come up later, or if you want to join his class that he's offering. That's so generous. Thank you.
Yeah, not kidding. Thanks again for this opportunity. It's good to see y'all.
Yeah. All right. Well, thank you everyone for attending. Our next meeting will be on April 8th from 9 to noon. And we look forward to seeing many of you there. Thanks for coming.